Good morning, everyone. A warm welcome to everyone for the third day of our Gyan workshop. To begin today's session, I would like to request Kavita ma'am to please deliver her first lecture. Thank you.
All right, so today we'll cover 4 lectures. So we are going to go in some really interesting topics about cancer. So this is epigenomics, and you guys heard a little bit about epigenetics yesterday. But after that, we will switch to biomarkers, a completely different topic. And after that, we'll switch to yet another completely different topic. So we have heard a lot about cancer and how devastating it is and all those features of cancer, right? Right now, we are looking at the mechanisms. Then we will go to actual targeting of cancer. Okay, and that's where it gets really interesting. So, so today we look at epigenomics and cancer in the current landscape. So you guys already know what are the epigenomic or epigenetic modifications, which mainly involves DNA methylation, histone modification, which could be acetylation, deacetylation, methylation. Non-coding RNAs, they play a very important role. And then chromatin remodeling. Chromatin structure, if it is open, more expression of genes. If it is closed, less expression of genes. Okay. So the thing is this, we were discussing about epigenetics. So when you make a compound, right, against DNA methylation, so DNA methylase enzymes are the one which are methylating DNA. So some are doing the good job, some are doing bad job, right? And so how do you target the wrong guy specifically without hurting the good guy, right? So it is a very easy way. Of course, you have to make a drug which is selective for that isoform. But look at all of these things. All of these things control epigenetics. So when we exercise, for example, we don't have to worry about that whether DNA methylase 1 or 2 or 3, which one is activated, which one is inhibited. Exercise just knows, your body knows which one to upregulate, which one to downregulate, which one to inhibit, which one to activate. All you are doing, or all we are doing, we are eating good food, less stress, exercise. Body knows what to do. We don't have to worry about it. We just forget about all the things. The body will take care of all the good things. Body will eliminate all the bad things. Okay? So we don't get exposed to toxic chemicals. So if someone is smoking, you don't go in that area, because secondhand smoke is sometimes or a lot of times worse than actual smoking. Okay, drug abuse, of course, is very bad. Okay, this financial status, this is also very important. We'll talk about this a little bit later, how it is important in medicine. Okay, we were just discussing alternative medicines, right? So alternative medicines and all of these things, and we all know stress plays a very important part. So we all have to learn to live without stress, okay, or dealing with it in a very positive manner. So we will talk about 3 main things: oncogene addiction, synthetic lethality, and immune modulation. How many of you know about oncogene addiction? Do you know what that means? Anyone? You don't know that, right? So what happens is that cancer cells get addicted to oncogenes. And what are oncogenes? Those are the genes which cause cancer. So they will be too attached to one major oncogene, right? So they will overexpress that one protein in huge amount, and that will take over the whole thing. So that is the main driver. So when you are driving a car, there is a main driver, then there are passengers. So the driver is the one which is driving the car and making all the decisions. Others are just passengers. They may be helping the driver, but they're not doing much. So when you want to target something, you want to identify the driver, the guy who is driving the whole disease. So cancer cells have weakness. They become addicted to one or two oncogenes. So if you could figure it out which one is that oncogene, you target that. Like BCR-ABL, CML is addicted to BCR-ABL, and that is why it is leading to CML, right? So if you identify the oncogene, then you can kill the cancer. Okay, so that is oncogene addiction. Synthetic lethality, I will give you the actual example today, and then we'll discuss immune modulation. So oncogene addiction with respect to epigenetics. So you already know epigenetic alterations like all of these can lock in oncogene expression. So there is a very famous or infamous oncogene called MYC, 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 right? So a lot of cancers are addicted to MYC. That means MYC levels are very, very high in this cancer. Okay, so if you target, yeah, oh, absolutely, sorry. So if you target MYC, you can kill this cancer. Okay, so they have MYC addiction. So why do they have MYC addiction? So MYC is present in very small amount in normal bodies, and it gets degraded very quickly. Okay, but MYC is highly expressed in cancer, highly expressed. So tell me some ways by which it can be highly expressed. Tell me any mechanism. Don't worry about even if you think you are wrong, just make it up. I want us to be very interactive so that you can think ahead how. So, remember I said gene amplification can occur. So the gene is duplicated, triplicated, and then you can have more levels. So that is one way, right? But that is not epigenetics. From epigenetic perspective, how can MYC be upregulated? Yesterday you guys answered many questions regarding epigenetics. So tell me, acetylation, more acetylation of that, more higher expression. All of you agree with me. Less DNA methylation of the promoter, higher activation, right? So that's exactly what it is from epigenetics perspective. So when MYC is overexpressed, it is due to mainly epigenetic alterations. Make sense? That is why MYC is highly expressed. So that's what they're saying. MYC amplification often coexists with epigenetic programs that increases its transcription. Okay, so that means if you inhibit these DNA, if you inhibit histone acetylases, in this case, so I did not tell you in detail, when histone acetylases acetylate something, these bromoproteins, they come and bind there, and that increases transcription. Okay, so if you inhibit this bed domain proteins, transcription will be inhibited and MYC levels will go down. Is it making sense? Okay, so epigenetic drugs may withdraw the fuel for the addiction. Okay, so this is MYC. So why cells become addicted to MYC? Look at that, how many cancer hallmarks are fed by MYC. Okay, when MYC is overexpressed, it evades growth suppression, it avoids immune destruction, uncontrolled proliferation, cellular metabolism is all deregulated, cell death doesn't occur, genomic instability, angiogenesis, invasion, inflammation, every single thing. Cancer requires, right? One guy is controlling these many pathways, and that is why it is very important to target MYC. So look at this MYC. So normal cells have a lot less MYC, cancer cells have a lot more MYC, and more MYC, more angiogenesis, they become independent of growth factors. So again, if this room is a cell, and just imagine that the growth factors are outside, right? Coming outside, and only when growth factor binds here, the signal comes to the nucleus and cells are supposed to grow. But if they become, because, so you can imagine, signal is coming and somewhere MYC is here, and the MYC is telling cells to grow. Now MYC is already too active, it doesn't need any signal. It is independent of that now, right? So it becomes growth factor independent, uncontrolled cell growth. And all the cancers are very smart. What is the biggest enemy for cancer? Immune system. So they shut down the immune system. Okay, that is why immunotherapy is so effective. Okay, just waking up the immune system. So second thing is synthetic lethality, and we already discussed it, but today I'll tell you with using a real example. So once again, just to remind you, if their normal cells have two genes for the same pathway, A and B, so if you shut down one, any one of them, this other pathway can take care of it, and cells don't die. So if you hit B, A can do the job. If you inhibit A, B can do the job. But in cancer cells, one of the pathways already shut down. So they depend entirely on the second pathway. And if you inhibit that pathway, normal cells will not care because they have A, but these cells will die, right? So you are specifically targeting cancer cells. So this is a real example, the most famous example. So remember we discussed that in many cancer, BRCA1 and BRCA2 proteins are missing. They are entirely deleted. So what happens is that what is the job of BRCA1, BRCA2? So this is HR repair, this is SSB repair. HR repair, so when DNA is damaged, right? So it could be single-stranded, DNA is double-stranded, we all know. So it could be single-stranded damage, this is good, okay? Sometimes both strands are damaged at the same time, that is bad, okay? So there are many different kinds of mechanism to fix single-stranded DNA and double-stranded DNA, right? So this is homologous repair, HR. So this is one way of repairing the damaged DNA, okay? And this is done by BRCA1, BRCA2. See, you can see this is double-stranded damage here. Then there is this protein called PARP, which fixes single-strand damage. So this strand is okay, but this strand is broken. So see, single-stranded damage. So SSB shows single-stranded base repair, okay? So one is for double strand, one is for single strand. But the double strand can always do the single strand. It's a piece of cake, okay? So basically they can do both. So what happens is that BRCA1, BRCA2 is absent in many cancer, okay? So if you inhibit PARP in normal cells, BRCA1, BRCA2 can also do this job, okay? So the cells survive, no problem. But because BRCA1, BRCA2 is not present in cancer cells, if you inhibit PARP here, the cells will die. So if you damage the DNA and then you give PARP inhibitors, the cells cannot repair the damaged DNA. Make sense? And the cells will die. So that is synthetic lethality. You induce the damage and then you inhibit the repair protein, okay? And what is the advantage of this therapy? The advantage is that you are not affecting the normal cells, right? Okay, so that was that. So we have covered oncogene addiction, we have covered synthetic lethality. The third is immune modulation with respect to epigenetics. Okay, so we are just still at epigenetics. So what epigenetics does in cancer, that they choose to suppress all the tumor suppressor. Anything which prevents tumor from growing, they suppress it. And what is the major mechanism of suppressing it? DNA methylation, correct? So if you use the inhibitors of DNA methylation, then tumor suppressors will be expressed. And if you express tumor suppressors, cancer cannot survive, right? So that is why, so one thing they do, so it makes complete sense that the tumor suppressors are suppressed in cancer by DNA methylation. That is not only thing they do. Remember, I just said that if you have a working immune system, cancer cannot stay there, right? So one of the things cancer cells do, so anytime we are infected with any foreign material, there are these cells which express these MSC receptors, this major histocompatibility receptor. They are different classes. Let's not go into the details. And what they do, they show. So suppose we are infected with a virus or anything, right? So the virus will be chopped up into small pieces, and those pieces are shown by these MSC class 1 receptors like this. And that is how the antibodies are built against those things. So when we make virus for coronavirus, for example, how did we make the vaccine? What was the starting point? The virus has, so virus structure is like that. It has these spike proteins. So they took out the spike protein, chopped it up, and then they built antibodies against those chopped things, right? So it is the same thing. So if you don't even have MSC receptors, they cannot present those chopped part to B cells to make antibodies. Is it making sense? So they make them disappear. They are not even expressed. So then immune system is silenced because basically it's not working, right? One of its job is to show the pieces so the antibody can be built. So if you add DNMT inhibitors, which are DNA methylase inhibitors, these things will show up again, okay? So similarly, epigenetic drugs can induce expression of endogenous retroviral elements, mimic viral infection, trigger interferon response, recruit immune cells. All of these things are silenced by cancer cells. This is a way to basically completely shut down the immune system. Immune system can get activated by many things, okay? Anything which activates the immune system, cancer cells, make sure those proteins are not expressed, okay? Okay, so this is shown here that these are different mechanisms. So MSC class 1, these things are silenced by DNA methylation. So if you use inhibitors of DNA methylases, you can express these proteins back again, okay? So this is different kinds of mechanisms. So see, this is MSC1, okay? So this is not expressed in normal, tumor cells don't let this express, but if you give the inhibitors, see down regulation of MSC molecules. But if you give DNMT inhibitors, DNA methylase inhibitors, they will be expressed again. Immune system will be activated. Okay, they affect the tumor microenvironment. How do they affect the tumor microenvironment? So in the tumor microenvironment, tumor cells will express such signals that the when immune system cells come to eat up the tumor, they will change these guys from enemy to friends. So instead of killing them, now they are supporting it, they are nurturing it. So they secrete a lot of factors like chemokines and different things, so that all the cells which are supposed to kill them, they become the friends there, okay? And then they will express VEGF. You know, VEGF is something which promotes angiogenesis, vascular endothelial growth factor. That is the major thing which supports angiogenesis, blood vessel formation for tumor to feed. And then we already discussed PD-1, PD-L1, right? So they also control the immune checkpoint. So these are the three things shown here. So how do you control oncogene addiction? You can, at the epigenetic level, You can use BET inhibitors or DNA methylase inhibitors. So once again, if you don't remember about bromodomain, so you all know acetylation increases, opens up the structure. So when acetylation occurs, this BET proteins, bromodomain proteins, they come and bind to acetylated histidines. Sorry, not acetylated, acetylated histones. Okay, they bind there, and that increases transcription. So if bromodomain proteins, these BET proteins do not bind, transcription is not enhanced just by acetylation. It can be enhanced, but they really help enhance transcription. So if you give BET inhibitors, they will not be, they will be shut down. Okay, so and synthetic lethality we already discussed, and so did we talk about immune modulation. Okay, so you already know about hypermethylation. Okay, so these are some of the very important targets. DNA methylase. This is a kind of DNA methylase, a very famous DNA methylase, EZH2. So what EZH2 does, it basically suppresses a lot of tumor suppressors. Okay, it is a methylase, so it will do the hypermethylation of the promoter regions so that these proteins are not expressed, these genes are not expressed. Okay, BRD4, remember? So BRD4, why BET inhibitors? So we just talked about how bad is MYC, right? So BRD proteins actually increase the expression of MYC proteins by using epigenetics. Okay, so look at this thing here. Okay, so this is EZH2. So EZH2 does a lot of different things. Okay, so EZH2 controls chromatin structure, cell cycle progression, epithelial to mesenchymal transition. Do you guys remember? Epithelial cells become mesenchymal, and mesenchymal cells are highly motile and highly invasive. So invasion occurs, metastasis occurs. Okay, so it activates oncogene, it promotes anti-tumor immunity, basically shutting down this thing. So this is a very important target for cancer, EZH2. Okay, and EZH2 see down-regulates MSC1 expression. Okay, so and it increases PDL1 expression. So all the bad things is done by one guy, EZH2. Okay, and it does all the things using epigenetics because it is a methylation enzyme. Okay, so HDACs, what are HDACs? Can you tell me what are HDAC? Deacetylase, right? So here is the thing, things are not always black and white, that hats when they acetylate, they only acetylate the good guys, sometimes they acetylate the bad guys also. So they can acetylate an oncogene, okay, and if that is acetylated, it is open up, okay, and there will be more expression of that. So if they acetylate an oncogene, what do you want to do? You want to deacetylate it, right? Or sometimes what they do, so think about it. So deacetylate is done by histone deacetylases, right? So why would we use histone deacetylase inhibitor? Under which condition would we prefer to use HDAC inhibitor? So if we are using HDAC inhibitor, that means acetylation should stay. Deacetylation should not occur. So for which proteins acetylation should stay? Tumor suppressors, very good. So in this case, to prevent tumor, we are going to give HDAC inhibitor, so the acetylation on tumor suppressor stays. Okay, but suppose it is doing an oncogene thing, then we will not give HDAC inhibitor because then it is bad for us. Okay, all right, and see this picture here. So this is a very good picture actually. So look at this these are obviously histones, right? DNA is wrapped around it. And TF stands for transcription factor. So this is acetylated. Look at BRD4 protein. Okay, so look at, this is RNA polymerase here. RNA polymerase is the one which makes the RNA. So if you have DNA and you put RNA polymerase, nothing will happen because the DNA is so tightly packed. So you need at least 20 different proteins to come together before RNA polymerase will even go and sit there. Okay, so it has to be opened. So see, when you mechanically open something, right, you have to distort it sometimes, something you have to do a lot of things to really open something up, right? So that's what cells have to do. So see, as this is acetylated here, BRD4 proteins are binding. It is acetylated here, they are binding here. Then BRD4 cannot interact with BRD4, so this mediator protein comes in. Okay, this binds here. So see, this whole structure is distorted. Okay, so once again, the example I was giving, suppose the DNA is running from here to here and you have to open it here. This is not open DNA, it's very, very compact DNA. So you are distorting it, like you are really distorting. So see the distortion here, that this part is binding here. All of these kind of actions just really opens up the DNA. Okay, so now you can imagine if you remove BRD4 protein, the structure will collapse. Okay, so that is why. So if you add BED inhibitors, this structure will just collapse. The transcription will be inhibited or activated. Inhibited, very good. Okay, so this is the same thing all summarized here. How many of you know about PROTACs? Has anyone heard about PROTACs? That is a very, very super exciting thing. Okay, so there are two ways to target a protein. Keyword is protein, right? So if it is an enzyme, you can use an inhibitor. Have you ever heard of the word undruggable proteins? Undruggable proteins, right? Why? So if it usually when it is an enzyme, it has an active site where the catalysis is taking place. But many proteins, they don't have an active site because they work by interacting with other proteins. So it's very hard to make a small molecule for these guys. Plus, some people talk by mouth, some people talk by touch. So these are proteins which are just binding other proteins, right? So if you inhibit even the activity, it doesn't matter. They will still touch other proteins and they will still do all the signaling. So in that case, how do you target this protein? You can inhibit their activity, but they can still bind other proteins and they will keep the signaling going. So how do you target them? How can you target all different parts of the protein? And this is pretty flat protein. It's not a pocket protein. Can anyone think of it? Very good. If you stop the expression, the problem is, so yeah, you definitely can do that, what you suggested. You stop their expression, absolutely. And you can target epigenetics by doing that, absolutely. But let's say the protein is still expressed. And can you do something at the protein level to make it disappear? You are 100% sure. Epigenetics is so powerful. Now you recognize it, right? You cannot remove, you obviously can change the DNA by CRISPRing it out, but that's very complicated. So you use epigenetics using small molecule. But I say the protein is still there. How can you make that guy disappear? How can you make the protein disappear? No, you don't want to use protease because then protease will digest everything, all the proteins, and the cells will die. Everything will die. So how can you specifically degrade one protein? How can you make one protein disappear out of the whole mix? Can you even make a protein disappear? I'm not even asking about disappearance. I'm asking about if you have a target like MYC, where the signaling always goes by touching each other, all different kinds of association, there is no pocket. How do you inhibit this protein? Because if it has an enzymatic site like kinases, BCR-ABL, no problem. You make a small molecule. You can do drug design. That is a piece of cake these days to do that, right? but something which does not even have a pocket. Every talk is by binding. What will you do for that protein? Very good. You can do RNA silings. It's perfect, right? Epigenetics, very good. RNA silings, outstanding. So you are targeting epigenetics, protein is not expressed. RNA is expressed, she is targeting shRNA, RNA is degraded. So you guys suggested two outstanding methods. First, let stop it before RNA is made. She said once RNA is made, we degrade it. Wonderful. Let's say something still made the protein. Now you have the protein sitting there, and you cannot make a small molecular inhibitor for that. It has no on-off switch. That would be very good if it is localization dependent thing. So for example, for the longest time, RAS protein could not be inhibited. Okay, so but RAS protein only works, you all know RAS, right? The famous RAS. The RAS protein only works if it is anchored to the membrane. So they were using your technique. If you don't let RAS go to the membrane, it's useless. So for 50 years, everybody was doing that. So that's a very good way. But this protein is not localized. I'm just giving you a hard time for you so that you think just a little bit more. Because see how many good things came out of it. You guys already have fixed before expression. When RNA is there, you have changed the localization. If the protein, the culprit guy is standing in front of you, what would you like to do? But the thing is that there are 20 different sites there. How will you inhibit something so which is so undruggable? You make that guy disappear. In interest of time, I'm giving you the answer. You want to degrade the whole protein instead of inhibiting it. There is a huge difference between inhibition and degradation. Okay, suppose somebody is very talkative. You shut the person by putting a rasgulla on that person's mouth, right? That person cannot talk, right? But the person is still there. And he or she can do hand gestures instead of talking. When we don't know the language, we say, okay, we are still doing that, right? But we don't even want that person to talk to anyone, not even using hand gestures. What do? You make the person disappear. Protein degradation. You can degrade the protein and make it disappear. And that's what Protax is. When I'll tell you the details of this, you will love this thing, this method. Okay, it's a very, very powerful method. It was developed by Craig Crews at Yale University. I saw when he published the first paper, and this thing took off like this. Okay, and now there are so many Protax which have crossed, which are in clinical trials. It is that powerful method. So it is extremely, extremely powerful method, and we all know the power of CRISPR. Okay, so this is just today we are going to talk about biomarkers in great details, and you will learn a lot of things. So I will skip this slide because I will. So biomarkers are the one which guide the therapy in many, many different ways. Okay, we'll talk about that in after this lecture. Okay, so what happens is that biomarkers will tell exactly what therapy to use, when to use, how much dose should be used. And suppose there are 10 people, okay, 10 people are there, they have exactly the same disease. Okay, biomarker can predict which patient will respond and which patient will not respond. Identical disease, okay, but your genetics is different from mine, so they will do a genetic testing. That too is a biomarkers based things, and it will tell you exactly which person will respond even before the therapy starts. So they are very powerful, but we'll come to that a little bit later. Okay, so this is just the combination strategy. So you can use epigenetic drug, but you don't have to use it alone. You can mix it with something else to make it more powerful. So suppose somebody has HER2 dependent cancer, so you can use HER2 dependent antibody, which is Herceptin, plus epigenetic marker, okay, epigenetic inhibitors, okay. Epigenetic drug can be combined with immunotherapy. You are shutting down the, you know, the pathways, you are activating tumor suppressors, but you are also activating the immune system at the same time. You can put epigenetic drugs with DNA damage agents and PARP inhibitors. So can you tell me why we have to use DNA damage and PARP inhibitor together? On the DNA damage. If there is no DNA damage, you don't even need PARP. So PARP has to be combined with DNA damage, right? Okay, and we already talked about synthetic lethality pairs, right? So so we can, so epigenetic drugs often work at low dose, okay? So we have to find the right dose. Toxicity is always an issue because, you know, normal cells will also be affected by this thing. So we have to figure out which patients, how selective, and we have to think about the toxicity so the cancer cells die, normal cells don't die, okay? So now I'm going to show you 3 cases, okay? So in case 1, this is a hematologic cancer. So DNMT inhibitors are used to resensitize AML to other agents, and you can, so here what happens, tumor suppressors are suppressed by DNA methylation. So here, if you use DNMT inhibitors, you can re-express the tumor suppressors, okay? In this thing, EZH2 is also a DNA methylase, I just told you. So EZH2 mutation positive tumor show dependence. So if you inhibit EZH2, tumor suppressors are activated, okay? This is very interesting, and this is a little bit complicated, so I put the picture here. So IDH is isocitrate dehydrogenase. You have studied that in citric acid cycle. Okay, this is one of the steps. So remember, alpha-ketoglutarate is formed in citric acid cycle. That is catalyzed by this guy. So what happens in that, in tumor cells, this protein IDH is mutated. So instead of forming alpha-ketoglutarate, it forms 2-hydroxyglutarate, 2-hydroxyglutarate. I thought, yeah, this is what it is. 2-HG is 2-hydroxyglutarate. And this is called an oncometabolite. Have you ever heard of this oncometabolite? You know metabolites, right? All the things in citric acid cycle, they are basically metabolites. Generic term, like we say small molecules, everything, or every small molecule which is made is a metabolite. Onco, anytime word onco comes, means cancer. So if 2-HG is formed, that can cause cancer, and the way it causes cancer is that, remember, if the cells are differentiated, they are good, and if they are not differentiated, that's very bad news, right? Cancer is very aggressive when they are not differentiated. So when this metabolite is formed, it inhibits differentiation, and if it inhibits differentiation, cancer is very aggressive. So makes sense? So if IDH is there, it will form 2-HG, which will inhibit differentiation, basically promoting cancer. Okay, so you don't want IDH. So you will use epigenetic drugs to stop the expression like you suggested. Make sense? All right. Okay, so this is what it is showing, that if IDH mutant is there, it will form 2-hydroxyglutarate, and that will block differentiation. So we, in this case, we don't want IDH1 protein there. Okay, so we would like to stop the expression using epigenetic drugs. Okay, so future directions in this field is that you have to, so now people, so in 2000, people were doing like the whole tumor biopsy, genomics or proteomics and things like that. Now we can easily do in a single cell, like it's very, very easy. So why single cell? Because tumor is very heterogeneous, right? So if you take the whole bulk of tumor, you will get too much information and you will not know from where it is originating. So now you can actually pick up one cell and look at the whole genome there, epigenome there, okay? So that's what it is. We need exact, precise thing. And then we will come to biomarkers. Is it already 1 hour? No, not yet. Yeah, we have some time. Okay. So, see, every time these biomarkers are coming up, and you must be wondering what they are. So, next whole lecture is on biomarkers. Okay. And then we can make selective degrader molecules, which are PROTACs. Okay. So, I will tell you about PROTACs not today, but probably tomorrow or day after tomorrow. Okay. So, selective degrader molecules, which will basically make the protein disappear, which we don't want. Okay. So epigenetic regulators are actionable molecular targets that provide precision therapy opportunities. See, now we are working at the level of a single protein or a single gene, right? We are not giving chemotherapy. Chemotherapy is what? If you have to explain chemotherapy to a high school student or to a person who doesn't know anything about biochemistry, how will you explain chemotherapy? That all cancer cells are dividing very fast? That's the first thing. It's uncontrolled. We give chemotherapy, it stops cell division. So cancer cells die. What cost? All normal cells also start to die, right? So that's chemotherapy in very small, simple words. So just imagine how bad is chemotherapy, because now you are finding that one MYC protein, you are finding this one EZH2, and you are targeting those by magic bullets, correct? So that is why you have to have very precise knowledge of whether this is really the case there, okay? And then, so if you match the target and biomarker is coming again, you will know that. And then you can use the combination therapy by targeting two pieces at once, okay? And you can reverse resistance, increase, basically activate the immune system, and create synthetic vulnerabilities. You know, somebody who is very strong, they may have some new vulnerability which we are not aware of, right? So you can find cancer cells fool everyone, but they develop their own vulnerabilities. And if we identify them, we target that part. Okay? All right, that was good. At least one thing was on time. So, what is the schedule? Do we wait here? Yeah, we wait here, right? Oh, thank you. That's great. Okay, that's good. That's very good. I'm I'm pretty sure you guys are going to, because this is really changing gears and looking at cancer from a different perspective, and that is biomarker. We all understand what biomarkers are, right? Something which tells us about something, right? But you will be surprised how many different kinds of biomarkers are there and how useful they are. Okay, so I could not find one slide which had all the biomarkers, because some people put these biomarkers, some are common, some add more, but then these are missing. So I put different pictures there, okay? And I will tell you about all of them. So I will tell you what is coming, because you guys don't even know what is coming. Okay, so lecture 9 is completely based on what are the different kinds of biomarkers and how they are used. Okay, then lecture 10 is about health disparity. Okay, if I ask you health disparity, if you have to explain that, what does it mean to you? Because I certainly did not understand what it meant. I did not understand the full extent of what that meant. So can anyone of you can explain what is health disparity? No, no. So health disparity, I will tell you, before I put together this, I'm more experienced than you guys, but even I did not understand the extent of health disparity. So my understanding of health disparity was that in US there are white Caucasian population, and that there is a African American population. So originally they came from Africa, and they are Americans too, right? So they have been living in the same country for generations and generations and generations and generations. but they have different, if these guys will have HER2 positive breast cancer, these will have TNBC, triple negative breast cancer. They have the same thing. These people will die, they, they will not survive, they will survive. Like, there is a lot of disparity between that. So my definition of health disparity is, because funding agencies give a lot of money to study health disparity. Why people of this descent are more receptive, of this descent are not receptive, right? So here we don't have that much diversity, but in US there is huge diversity because of many different countries. Like Asian population, they show some phenotype, but Caucasian will show something and African will show something. So people have very different response. Okay, some people will have uh, the worst kind of cancer, same prostate cancer, but the worst cancer. But these guys will not have that kind of cancer. So my understanding, that is health disparity. That is definitely health disparity, but that is the only thing I knew about that. So we'll discuss in the next lecture what exactly is health disparity. And then in the last lecture of today, we'll come back to explain that how you could use biomarkers to identify and fix the health disparity. So that's how the whole structure is arranged. So let's start with biomarkers there. Okay, so as you can see, there are many different biomarkers. So what is a biomarker? Biomarkers are measurable so biomarker could be anything which tells you about the state of a system, that is a biomarker. It could be a DNA, like it says here. It could be a molecule, like a metabolite. It could be a gene, protein, imaging feature that provides valuable information about health and disease. Okay, so it could be a receptor which is expressed. In some cases, that is a biomarker, like HER2 positive. HER2 is a biomarker because it is telling you it is HER2 positive cancer, right? If, like prostate cancer, PSMA is a protein which is expressed at high level, that is a biomarker, right? So this is a normal individual with normal biomarker, but see, here it is expressed in huge amount. So now it becomes a biomarker, all right? So how do you find, so biomarker can be a DNA, it could be a RNA, it could be a protein, it could be a metabolite. And so how do you find it about DNA? So suppose my DNA is different, some part of my DNA is different from, I don't even want to say my, because I don't want to even associate my disease, me to disease or you to disease. So we'll talk in abstract terms, okay? A and B, normal and unhealthy. So how do you figure out if the problem is at the DNA state, right? So some DNA has changed in this one, which is leading to disease. So now we have to profile the DNA, because if we look at the RNA, it's not going to help. We have to profile at the DNA. Many times DNA is perfect, same. This is the diseased guy, this is the normal guy. DNA is exactly the same. The protein is the problem. Okay. Even if you have the same DNA, the protein can be different. So if you have to figure out, now if you are doing DNA profiling, it's not going to help. Now you have to do proteomics, right? So that is why if you have to do RNA, you do microarrays. So for DNA, you use FISH, F-I-S-H technique. For RNA, you use microarray or gene chip. For proteins, you use proteomics. For metabolites, you use metabolomics. Okay, so that is what they are showing, that you have to use different techniques to find out at where is the problem, what is the biomarker which is different between normal and disease state. Okay, so this is FISH technique, just in case you don't know. So you take the DNA from normal and diseased patient, you separate the two strands together. Many of you have done PCR, denaturing, heat it up, as simple as that. And then once you denature it, you label them with probes, and then so these probes are denatured, and then You take a known sequence of DNA and see if that probe binds there, and that will tell you whether it is binding or not binding, and that is how you can discover the abnormality in DNA. Okay, so this is that thing, and this is chip. So anyone knows how RNA chips work? No. So I will tell you the story during the break about RNA chip, how it was discovered. It is a very interesting thing. Like five real smart people sit together and how this technology came together. So I know the inside story because one of the person who developed this chip was the director of the institute where I worked. Okay, so so what you do is that this is normal, this is diseased. So you take out the tissue, the biopsy tissue, normal tissue, normal prostate, diseased prostate. Okay, and then you make the RNA out of that, so, which is a very easy thing. You make the RNA from this one, RNA from this one. The RNA from this one, you color it green. RNA from this one, you color it red. So you're not literally coloring it, you're attaching a probe which will give red fluorescence versus green fluorescence. All of these are piece of cake, okay? And then you make a chip where DNA sequences are there, small DNA sequences. So suppose you want to check 20 genes, suppose you want to check 100 genes, suppose you want to check 200 genes. Suppose you want to check 5,000 genes. So you make a DNA chip and you will have unique sequences, 20-mer sequences of those unique genes. So for example, if you're comparing gene 1 to gene 2, even if they're close, something will be unique about gene 1 versus gene 2. So you always use unique sequences because you're not using the whole gene, you're using just a small piece. So that small piece should be enough to tell you this is for that gene, this is for that gene. So you have a chip which has the DNA sequence there for all different genes, small pieces in each well. Make sense? Now you add the RNA. RNA and DNA will hybridize if there is exact complementary. So DNA sequences are the complementary sequences. RNA, if it binds, signal will light up. If it doesn't bind, signal will not light up. Are you guys with me here? And then you compare. So here what they have done, they have mixed it together. So they took normal cells, disease cells. They mixed the RNA together. One is green, one is red. Correct? Then they add the mixture to the chip. So if both binds green and red, the color will be yellow. If only green binds, it will be green. If only red binds, it will be red. So now look at this thing. Pathological cells are red. Green are normal cells and yellow when both are present. So there is one chip which will tell you these genes are okay because they are expressed in all. Yellow ones are good. Both are having. Green ones are only present in normal cells. That means red cells don't have that RNA. Bad news. And then red cells have something which even normal cells don't have. That too is bad news. So one chip has given you so much information, right? That is gene chip. Okay, very simple, right? Okay, I will tell you a story about gene chip in our lab also. Not in my lab, but when I was a student in that lab. That was before this was discovered. Okay, we were playing a game and there was an undergraduate in our lab. Okay, maybe 17, 18 years old. She suggested this method. Okay, and that time nobody had ever done the green color and the red color. And she said, you make one green, one red, you mix them together. And I remember I was laughing because I said, how can you make it one red and one green? Because I could not think that you could use a probe. But she said, if you could make one red, one green and mix them together, this will work like that. So this idea came from an undergraduate. So the point here is that think wild. If you can think it, you can do it. You know? Okay, so biomarkers now we know can be DNA based, could be protein based, could be also lipid based because lipids are very important. It can be sugar based. We are not talking about glycosylation in this course. Have you guys heard of glycosylation, right? So all the many proteins in the cell, they are glycosylated. And glycosylation pattern differs between normal cells and pathological cells. So you can even find what kind of pattern is present, and that could also be a biomarker. Okay, and you can do tons and tons and tons of things with the biomarkers. You can identify exactly what kind of cancer it is. Okay, HER2 positive, HER2 negative, triple positive, right? You can monitor the disease progression. You can monitor whether it is getting better, whether it is getting worse. You can tell how severe it is or how benign it is. You can do screening of drugs. You can predict treatment response. You can predict what drug you should give, and we'll come to all of this in just a minute. So these are some of the types of biomarkers, like predictive, prognostic, diagnostic. Let's go one by one. Okay, so this slide I just put this morning here because this was not supposed to be there, but I thought that if I tell you about this biomarker, then this biomarker, after 2 or 3 or 4, you will get all mixed up. So I thought I will start with the differences, then I will go over it, and then we'll again come to the differences so that you really understand the difference, because sometimes it seems they're overlapping with each other. Okay, so look at some of the biomarkers here. Some of them are very easy to follow. So the risk is that, what is the meaning of risk biomarkers? So all of us are normal people, thankfully, on this day, but who is more prone to develop some sort of cancer? That is the risk biomarker. Disease is not yet there. We are just predicting that is there a possibility that somebody might develop develop a disease. So who is likely to develop a disease? Is it dependent on treatment? No, the disease is not even here, so there's no treatment there, right? So this is before disease. So then we can do prevention screening. A very good examples are that women, they undergo mammography. It is a must thing to do every year in US, because so many people's life have been saved, because and the insurance company do not charge for that. If you charge for it, half of the people are not going to get it done. So they make sure that is free once a year. Okay, and so numerous people have been saved by that one, because when there is no disease indication, but it is a possible risk there, especially for women after 40 years old. Okay. Okay, diagnostic. Does the patient have the disease? Okay, so sometimes there is a pain in the stomach and we don't even know whether it is just a simple pain because we ate something or is something serious. So diagnosis is that we have to find out if the disease is there or not. That is diagnosis, right? We know the meaning of diagnosis. Treatment dependent? No, because we are just diagnosing the disease. Okay, so we are suspicious that because the stomach is hurting, is there a disease, and we are diagnosing it. So this is basically disease detection. Okay, prognostic. Prognostic is that this guy has triple negative breast cancer. Oh, that is very severe. That's a very bad news. Oh, this guy has HER2 positive. No problem. This can be treated. So we are predicting the future here. We are prognosis. Pancreatic cancer prognosis is extremely bad every time. Breast cancer, not at all bad. Testicular cancer, somebody said to me, somebody has to have the cancer. They are forced to have testicular cancer because it's just so easy to repair. There is another cancer, somebody was telling me 2 days ago, I don't know who was telling me, and they said that is the best cancer to have because it's 100% treatable kind of thing. So we are giving prognosis, right? This is bad, this is good, this is... Cancer is bad, obviously, but different cancer have different kinds of prognosis. That's what prognosis means. How the disease will behave. Treatment? No, we are just telling pancreatic cancer is bad, right? So no. So this is after diagnosis. We are predicting the outcome. Okay. Predictive. When you say predictive, it is different from prognostic, because now say, okay, this is HER2 dependent cancer and we are going to give Herceptin. So we are predicting, yeah, it will work because it's HER2 positive, Herceptin should work. So it is predictive biomarker. So this is we are planning before therapy. We are selecting it for treatment based on what is wrong there. Pharmacodynamic, okay, this is very important one. So what happens is that you have given the therapy, but then you are monitoring, is it really hitting the target, is it going down? That is pharmacodynamic. We'll spend a lot of time on this biomarker. Okay, and then therapeutic, what is the drug's molecular target? To find the exact mechanism of action, again I will tell you the details about that, and there are even more. Okay, so risk biomarker indicates susceptibility. They are used in healthy individuals. BRCA1, BRCA2, Angelina Jolie, no cancer. She got she got all the surgery so that she never has those cancer. So it does not diagnose disease. It is just telling you that these people are at risk of breast cancer. Diagnostic biomarker, you actually say whether the disease is present or not present. We are diagnosing the disease. Okay, what is the advantage? Aid in early detection of the disease. Most of the cancer, if you can detect it early, you can treat them. Pancreatic cancer is so lethal because you cannot detect it in stage 1, stage 2, stage 3. It is, can only be detected in stage 4. And by that time, it is too late. Okay, so for prostate cancer, people do PSA test. For breast cancer, they do HER2 test. Okay, so this is diagnosis. This is just telling you some more diagnostic. So prostate cancer also has BRCA2 deletion. Colorectal cancers have KRAS mutation, EGFR mutation. So all different cancers have different kinds of markers, and you can use that to diagnose what kind of cancer it is. Obviously, if it is in the colorectal region, it is colorectal cancer, but that is not enough. Just knowing one thing is not enough, so you can tell exactly what kind of cancer it is. Prognostic marker, I told you in the very first class, tumor stage, TNM staging. More zeros, better it is. Higher the number, bad it is. That's prognosis. Higher number, oh, it has reached lymph nodes, poor prognosis. It has not reached lymph nodes, good prognosis. No metastasis, good. Metastasis, as bad as it gets, right? That is all prognosis. Okay, how aggressive it is. Predictive biomarkers, now we are coming to therapy. So predictive biomarkers will say treatment, predict response to a specific therapy. For example, if you look here, EGFR is mutated in, this cancer here, and also EGFR, this one, colorectal cancer, right? So if it is EGFR, so you can predict what kind of therapy one should be given for EGFR. Obviously, we have to target EGFR. So we will use EGFR inhibitors. If it is HER2, this is another name for Herceptin, right? So Herceptin, if it is PD-L1, immunotherapy. So this is the question, which drug should I use? That is predictive biomarker, okay? So this is more about predictive biomarker. So EGFR mutations in lung cancer. Huh? 10:30. Okay, can I just finish this slide? Thank you. Yeah. So I'll stop at this slide, okay? So predictive biomarker will tell us exactly based on what kind of, like if HER2 is amplified, we will use Herceptin. BRCA1, we will use PARP inhibitors, right? So that plays a role in patient selection and precision oncology. And you could give precise drug to that patient. I will stop here, okay? All right. What will we do? Oh, tea only. I heard something else also. अभी शुरू कर दिया क्या? अभी एक मिनट, अभी शुरू मत कीजिएगा. What was our plan? How did we go yesterday? Like after the second lecture, we take another 5-10 minutes break, right? Before we go, but we don't have any tea break after that. So we just take 10 minutes break and then we go for the third and then we go for lunch. Then we have one more. Then we have tutorial. Okay. All right. Okay, so now we come to predictive biomarkers. Oh, I already told you about predictive biomarkers. So the next one is therapeutic biomarker. Okay, just 1 minute. Okay. Okay. So therapeutic biomarker tells you about the mechanism of action. So what it tells you, what is the drug targeting? So for example, for imatinib, right, Gleevec, this is the another name of imatinib, it is targeting BCL-ABL. Nivolumab is targeting PD-1. Okay, so therapeutic biomarker tells you about the molecular target of the drug itself. Then pharmacodynamic biomarkers are extremely important. Okay, they monitor how the drug is behaving in the body. Okay, so that markers measured biological changes induced by the drug. Okay, whether the drug hit its target and produced its intended biological effect. So it confirms the mechanism of action, validate target engagement, and that also tells you what dose you should use. So in tomorrow's lecture, we will talk about the dose in great details. Okay, so for example, if you give HDAC inhibitors, so if you give HDAC inhibitors, acetylation should increase or decrease. HDACs are the one which remove acetyl group. So if you are inhibiting HDAC, acetylation should increase. So we are giving a patient HDAC inhibitor, and then what we will do, we'll check if acetylation levels are increasing. If it is increasing, you know the drug is hitting the HDAC, right? So similarly, if you are giving angiogenesis inhibitor, that is anti-VEGF drugs, because VEGF increases angiogenesis, then you will look at VEGF signaling, right? So those will be pharmacodynamic biomarker. In simple word, it shows what this drug is doing to the body. Okay, so these pharmacodynamic biomarkers, in abbreviated form, they're called PD markers. They are very important in drug development. So that confirms the mechanism of action, that this is the mechanism of action. See, think about a disease. Disease is very complex, extremely complex. Okay, so you are predicting that this is the thing I should give to this patient. But then how do you know it is even working? If that is the thing. So then you have to confirm it, right? So that is where pharmacodynamic biomarker comes in it. So by measuring the functional consequences of a drug's interaction with its target, they can confirm that the drug is having the desired effect, what you want to do. If it is still not working, that means that may not be the main reason why the person has this disease. So that is why you have to maintain that thing. Suppose you are giving an anti-cancer drug, right? So what is the end result of anti-cancer cells? Cancer cells should die, right? So one of the way to measure cancer cells is that they are replicating. So there is a marker called Ki-67. So if Ki-67 levels decrease, that means drug is effective. Sometimes you cannot see the tumor shrinking right away, but if the drug Ki-67 levels are going, you know your drug is working because proliferation is going down, right? So that is what it means, okay? So that is very important for in clinical trials to see whether the drug is actually working. And you can actually, based on PD biomarkers, you can improve efficacy. This is working, this is not working, what dose is working. You can make a lot of decisions there. and you can reduce toxicity and increase efficacy. Okay, and PD biomarkers also help in understanding resistance. So resistance is one of the biggest thing in cancer. You give a drug, you are happy about it, but then cancer cells develop resistance. So initially it is working, later on it is not working. So in the last lecture of today, I will show you an actual graph. You will be so happy to see, oh my God, this biomarker is decreasing like this. And then you see, and it is increasing again. So now you know it has developed resistance, right? So PD biomarkers are very important. Okay. So here I'm comparing predictive, prognostic, and pharmacodynamic biomarkers. So predict, identify patients which are likely to benefit from a specific therapy. Prognostic will tell you the outcome. We are making an outcome: pancreatic cancer, bad prognosis; breast cancer, good prognosis relatively. Pharmacodynamic will tell you the biological response to a therapy. See, look at this. Is it really hitting the target? That's what it is. Okay. So there is a comparison table: predictive, therapeutic, and pharmacodynamic marker. Predictive is before treatment because you're trying to decide what drug should be given, right? Therapeutic biomarker is during treatment, and this is shortly after drug exposure. Is Ki-67 going down, right? This is more about, is it hitting the right target? Okay. This will predict benefit and non-benefit before treatment for predictive. Which patients are going to be benefits? For example, it is HER2 cancer only, and you're giving Herceptin, it will be only affected for HER2 cancer, not for any other cancer, right? So that's what it is telling. It will help you in treatment selection. It will tell you whether they are going to be sensitive or resistant. So somebody has HER2, but you are giving a drug for BCR-ABL, it's not going to work, right? So you have to have the right patient for the right drug here, okay? So, and so this is some examples: BRCA1, EGFR. If BRCA1 is intact, you are not going to target olaparib. You are not going to give PARP inhibitors because they're not going to respond to PARP inhibitors, right? So that's all covered predictive. So therapeutic biomarker tells you, is the therapy working? If the therapy is not working, you could change the dose of the therapy. Okay. So, and it will tell you PSA levels are going down. Therapy is working in prostate cancer. Okay. BCR-ABL is declining. Gleevec is working. If BCR levels are not declining, BCR-ABL is not working. Okay. All right. Pharmacodynamic biomarker, like I said, Ki-67 is a very good. Ki-67 levels are going down, cancer cells are less and less. Okay. If you are giving HDAC inhibitor, histone acetylation should increase. So that is all pharmacodynamic biomarker. So I will show you many case studies, and these are very interesting to see case studies. Okay. So case study for diagnostic biomarker. So this is a prostate cancer case where it is PSA. We measure PSA levels. So the biomarker is PSA levels in serums. In prostate cancer, PSA levels increase. So many people who are over 65 or 70 years of age, they undergo PSA test regularly. Okay, because many times, even before the cancer is actually visible or identified, if PSA levels are rising, they will immediately get it tested. Many times men have enlarged prostate, but they don't have cancer. So to make sure they don't have cancer, you measure PSA levels. Okay, so that would be a diagnostic test. Elevated PSA levels suggest possible prostate cancer. Okay, but it could also be elevated at a very initial stage before cancer actually develops, but it will give you a warning, some red signal. Okay, so case example: a 65-year-old man, no symptoms, elevated PSA routine test, just the annual physical exam. Prostate biopsy confirms localized prostate adenocarcinoma. No symptom, no pain, nothing, just PSA levels told. What did you do? Surgery, gone forever. Okay. Case study 2, HER2 expression in breast cancer. So remember, mammography is a part of the annual physical exam. So 48-year-old woman with palpable breast lump. So, so in mammography, they will find if there's some lump there. And once they find, they looked at HER2 gene by FISH. You know, FISH will tell you if the gene is amplified, or protein expression by ISC. Do you guys know what is ISC? You know, immunohistochemistry. So you take the tissue sample, and when you are looking in the tissue, you see, you call it histo. If you are looking in a cell, you call it cellular, right? And so it is antibody based. You add the antibody, if the signal lights up, protein is there, right? So it's immunohistochemistry. So that's how this case, HER2 positive breast cancer, was identified. So see, the person just went for mammography, and they found that this person has cancer. So biopsy confirms this thing. This was the score, HER2 positive. This, the patient receives Herceptin. Okay. Now, case study 3 for diagnostic biomarker, pancreatic cancer. It doesn't show up. Okay, because there is no biomarker for stage 1, stage 2, stage 3, because it is almost, it shows rarely any symptom there. So there is a big gap in our understanding about pancreatic cancer, because we don't have the biomarker to detect it at the early stage, because it doesn't even show any symptoms at that stage. So that's a very bad news, and that is why pancreatic cancer is the most lethal cancer, no doubt about it. But the good news is that very few patients get pancreatic cancer. Okay, that is the only good thing about it. Like lung cancer, huge number of people get lung cancer. Okay, so unspecific symptoms, there is no screening, there is no biomarker, early stage, you don't even have imaging thing. You can image it, but you don't know what you are looking for. So you can look at the pancreas, but then what is the abnormality we are looking at? So we don't even know what to look at, right? So there is a huge gap in biomarkers in pancreatic cancer, diagnostic biomarker. We cannot diagnose pancreatic cancer at early stages, right? And that makes this cancer lethal. Okay, so gaps in diagnosis. We don't have any, we don't know what exactly we are supposed to do to stop it, because we don't even know how to diagnose it. Forget about stop it, we don't even know whether this person has it or doesn't have it. Okay, now let's look at prognostic biomarker, p53 mutation in colorectal cancer. If somebody comes and tells you this person has p53 negative breast cancer or prostate cancer or whatever cancer, what would you think first? What is your first thought? So suppose somebody comes to you, that person A is diagnosed with this cancer and it is p53 negative. Huh? And if it is negative, then what is your prognosis? Bad. Yeah, and it is completely gone. It's bad news, because tumor suppressor like p53 disappearing is not good news, right? So you can say, if somebody comes, this guy is HER2 positive breast cancer versus triple negative, triple negative is very hard to treat, your prognosis would be very bad for triple negative, right? If somebody says pancreatic cancer, pretty bad prognosis, right? So that's what prognostic biomarkers, we are predicting the outcome for that patient. Okay, so p53 gene mutations means aggressive tumor, right? So now we have to really closely monitor the patient. Okay, another case, HER2 in breast cancer. HER2 is overexpressed. So before we developed this drug Herceptin, what are the things I'm telling you? These are very recent discoveries, because we have made tremendous progress in cancer biology in the last 20 years or so, maybe 30 years. So especially CAR T therapy and all these things, these are all, actually less than 5 years. So, so before that, it was just across the board chemotherapy, chemotherapy, surgery, radiation therapy. So it was pretty bad. So that's what it is saying. Before, this is the trade name for Herceptin. So before Herceptin era, HER2 positivity was poor prognostic marker. People used to die. It was highly fatal disease. But with Herceptin, prognosis has improved. Okay, so it HER2 positive status alerts oncologists to more aggressive tumor biology. It is still better than triple negative breast cancer. Okay, and we have a very good drug, Herceptin, against HER2 positive one. Okay, so now let's go look at the case studies of predictive biomarkers. A 54-year-old woman presents with metastatic non-small cell lung cancer. A tumor biopsy performed, followed by immunohistory, immunohistochemistry, and next generation sequencing. Now they are telling you, they are presenting you the whole case. You guys have enough background in cancer biology. I will ask for your prediction. Okay, so this one person has this thing here. So PDL1 is expressed. Okay, okay, tumor mutational burden low. Tumor mutational burden means that the tumors are undergoing mutations all the time, so that they survival of the fittest for that. Okay, EGFR is exon 19 deletion. So you don't know what is this. When EGFR, this, you know what are exons? Do you guys know what are exons? You don't know that. You don't know that. You guys don't know that either. Okay, even if one person doesn't know, it's the same thing whether two or one. Okay, so what happens is that in E. coli or any prokaryote, prokaryote and eukaryotes, right? So all prokaryotes, the DNA is there, and the RNA is exactly like DNA. So if this is one gene, then the RNA will be made like this. Okay, and then RNA will make the protein. So suppose I go to RNA is identical to the DNA, but in eukaryotes, that is not the case. So when RNA is made, it has different regions, exon, introns, exons, introns, exon, introns. So then all the introns are deleted and all the exons are ligated. That's the mature RNA. This process is called splicing. And then this mature RNA will make the protein. Is it making sense? So the DNA sequence will make the RNA with lots of introns. Introns are diluted, exons are ligated. So you would think, why nature has such a mechanism? What is the advantage? The advantage is this, that this is the DNA sequence, right? And you make this huge RNA with a lot of introns. So in many, many cases, these introns are not ligated exactly at the same way. Now you have so many pieces, 1, 2, 3, 4, 5, 6, 7 exons. So these 7 exons in one tissue, remember every cell in our body has exactly the same DNA, but they have different RNAs. They have some housekeeping genes which are similar in every cell of our body, like glycolysis genes, right? Krebs cycle genes and things like that. But then they have unique genes also, because neurons are different from intestine, from skin. Liver cells have different function, neurons have different functions. So they obviously have different proteins. If they have different proteins, they have different RNA. So for example, so one DNA RNA is formed. Now let's say it has 7 exons. So these 7 exon containing RNA is spliced differently in the brain to make a different protein, but the same 7 exon RNA is spliced in a different pattern in thyroid, and here it will make a different protein. So you have the same DNA sequence, but they are 2 different proteins. So one such example is that there is one RNA in thyroid. It forms calcitonin, and calcitonin is involved in controlling calcium levels here. But the same RNA is spliced differently here, and it forms CGRP protein that is involved in pain signaling in migraine. Okay, so now nature has increased diversity at RNA level, right? So those are exons. Some exons are deleted, some exons are ligated, and the ordering can also change. So you can actually, but it is a very controlled process. It's not random at all. But the point is that from one DNA sequence, you are making different one. So in this case, so this obviously has many exons because exon 19 is deleted. So what is the consequence of exon 19 deletion? This EGFR protein becomes very active. So EGFR is a receptor protein, okay, which has an internal domain. It is a kinase. So it's a huge protein. So imagine that the cell membrane is this one here. So this is the part which is outside the cell, and this is the part which is inside the cell. Okay, so this is one EGFR, this is another EGFR. It binds a ligands, and the ligand is common, so the two things come together. So these two domains come together, they phosphorylate each other, they become active. But by deleting that 19 exon, one piece of it, this is always active. It doesn't need EGF, so obviously it is a very bad news because it's super active. That's what positive is the result. KRAS, if it is hyperexpressed, it's a very bad news because it leads to cancer. But it is ALK, so this is wild type, it is not mutated, good news. KRAS is present in the cells as long as it is not mutated, it's good news. ALK rearrangement negative, MET is a protein, is a kinase, it is not amplified negative, good news. So these two, ALK is not negative, so we can predict, we cannot use ALK inhibitors because that is not even a problem. KRAS, MET amplification negative, of course we will not use MET inhibitors for this case. PDL1 is expressed. Will you use immunotherapy? Of course we will use immunotherapy. So we know we have to use immunotherapy against PD1, PDL1. Okay, predicts, this is something we have not covered, but tumor mutational burden, if it is low, predicts weaker immunotherapy benefit. But still PDL1 is very important and it is positive, so we will still use PDL1. Okay, exon 19 deletion, we definitely will use EGFR inhibitors. Okay, because that is a major thing. KRAS wild type, if it is mutation, then EGFR therapy will not work. Absence will tell us which is absent because here it is no mutation, it is wild type. We know EGFR therapy will work. So by looking at the biopsy results, now we have decided the drug. Makes sense? That is predictive biomarker. And you can see how important it is. Before starting, we are not giving chemotherapy. We are personalizing it for that one patient, right? All right. So this is the details of what I just told you. This is worth going over. EGFR deleted means EGFR protein is very active because only a small portion which regulates EGFR is deleted, so it is super active. We will use EGFR inhibitor. PDL1, of course, we will use immunotherapy. Okay, so we have decided, and we give this drug, and after 12 weeks, the outcome is tumor has actually shrunk. Guess what? If some cancer patient hears that tumor has not, the tumor is stabilized, that itself is a cause for celebration, that it is not growing. That itself is a big cause for celebration. That tumor is shrinking, it's fantastic news after 12 weeks. Okay, so this is good. So basically we have predicted based on the biopsy results. Case study 2, Ki67 is a proliferation marker. Remember? So high Ki67, bad tumor. Low, so high Ki67 indicates rapidly dividing cells, disease is aggressive. Patient receives closer follow-up. We have to really monitor this patient very, very closely. Okay, now let's go to pharmacodynamic biomarker. Case study 1, imatinib. This is Gleevec in CML. So a 48-year-old man with CML starts on imatinib, and then it is very easy. You can do PCR. So imatinib, what it does is that, so I had to check this thing because if BCR protein is there and imatinib binds BCR protein and switches off the activity, right? So the protein activity is low. So why would you do PCR to look at PCR levels? Because when you do, you guys all know PCR, right? PCR, you are looking at what levels? You are doing qRT-PCR basically. So you are making the RNA out of it and you are measuring the RNA levels, that how much RNA is there, right? Transcription level. So if you're inhibiting the protein, why would you look at its transcription? And the reason is that In many cases, suppose I'm inhibiting an enzyme. So the real test I would do is to isolate the enzyme and see if it is active or not active, right? Why would I look at the mRNA level? I'm not touching the mRNA level. But in the case of BCR-ABL, it is slightly different. If BCR-ABL is inhibited, then BCR transcription goes down. And the reason its transcription goes down, it's like BCR-ABL is standing here and it turns on the pathway so that it is produced more. So it activates the signaling to make more of itself via transcription. So that is why BCR-ABL is inhibited, even the levels go down because the transcription of BCR-ABL goes down. That's why you can do a quick PCR. How easy it is to do PCR? 10 minutes, 1 hour later, you know it, right? When COVID was at a rise, people were doing COVID-based tests, right, by PCR. They would take a swab, and 2 hours later, you had the result, whether you have it or you don't have it, right? So, so you do QRT PCR there, right? And that will tell you. So in 3 months, BCR-ABL transcripts dropped by more than 90%. You know, so pharmacodynamic biomarker said disease is really, really working, right? Okay, take home message in biomarkers. Biomarkers are measurable indicators of biological process. Again, I'm just showing you 4 here, but there are more than 4, right? So now you can understand how important are these biomarkers of cancer. And they can really help you with the right therapy and everything. Okay, so what are the challenges? We still don't have reliable biomarkers for many cancer. Pancreatic cancer is one of them. And the biomarker needs to be very specific, because if it is non-specific, it is a useless thing, right? You have to have, so we have to integrate genomics, proteomics, metabolomics. And when you are, you know, when you have to measure the biopsy, if something is at the surface, it is easy to biopsy. But if it is an internal organ, you don't want to go there and, do a surgery to take it out. So many times what happens that its tumor cells shed something in the bloodstream, and you can just withdraw blood and you can see if those markers are there. Try to make it as less invasive as possible, not to stress the patient. Okay, so that biomarkers can really help us make the right clinical decision to treat that patient. Okay, wow, good. So let's start the third one till we are given the signal to stop. Okay. So after telling you about the biomarker, the next thing, before continuing about the biomarker, I will tell you about the health disparity. And after the health disparity, we'll come back once again to the biomarkers. Okay. And why this is okay. So why? So we have to understand what is health disparity first. Okay. So once we understand that, then how can we use biomarker to reduce health disparity? So this picture is really nice here. Okay. So look at these people here and look at these people here. Okay. So these people have access to advanced access and facility. So they can get the screening done. They are presumably pretty either powerful people or important people or rich people who have access to all sorts of things. And they are living in an environment where they have access to all of these things. Because you could be living in an environment which do not even have these facilities. If you're living in a rural village, they are not going to have all these facilities, right? So they appeared more educated and more, affluent here. And these are the people, they have limited access. That is health disparity. Okay, so it is not what we were saying again, that these people have aggressive disease. That is also health disparity, but this is also health disparity, unequal access to health care, health disparity. Okay, Why these people have access, why these people do not have access. That is health disparity also. Okay, so this is precision medicine divide, but this is not the only thing. That is the important part. So usually underserved population often face barriers to accessing precision medicine. This sentence is very loaded. So what is precision medicine? That if someone has a disease, you give exactly the right drug to the patient. Okay, that is precision medicine. But if the poor patient does not even have access to the actual screening process to figure out what is going on, I will tell you case studies, and these case studies are very important in with respect to precision medicine health disparity, because people don't know, they don't have enough money to get the screening. So somebody had HER2 positive cancer, this is a true story. So they did not have access to screen HER2, which will probably take 1 day, because you can do a PCR for HER2, but they did not. So what they were treated with? Chemotherapy. And the person died because, oh, this person has cancer, give chemotherapy. But a rich person comes in, they do the screening, they give HER2, that person survives. Right? That is health disparity. Okay? So I will tell you many things there. Okay? And all of these things are very important. Somehow there is a disconnection between the computer and this one. It shows something else here and something else here. So, so look at this picture here. So these guys, they have tailored therapies, real access to everything. And these are the guys, limited access. They are living in poor neighborhood with less access to stuff. So they don't get access to health service, right? So there are 4 things we will discuss here. Okay, precision medicine, you know already, but just a few sentences. This is the main thing. Precision medicine disparities, and then how we can identify actionable biomarkers. Actionable words is very uh, commonly used these days. Like if you find a good target, you call actionable target. Like if you take an action, it's going to show a result. That's what it means simply. Okay. Eliminating precision medicine disparity, then we combine the two things. We combine these two to eliminate precision medicine disparities by identifying actionable biomarkers. That is the other lecture of today, the last lecture of today. So precision medicine you already know so very nicely by now, right? And this is actually, precision medicine depends on a lot of factors, even where you live, in deciding that. Okay, the person's tumor genome and our own genome. So here is the thing. Suppose there are 10 people with 10, and they have identical disease, identical grading, identical everything, and you give them the same therapy. This person will respond differently than this, than this, than this, than this, than this, because their genetics is different. And because their genetics is different, they will respond definitely. So you have to look at the genetic also. The tumor the tumor genetic is different from the inherent genetic that we got from our parents. So we have to look at many factors before we come to precision medicine. As you will know more and more about stuff, you will understand this figure better. Okay, so we have to look at the immune thing, we have to look at the environmental exposures. Suppose somebody is living, so this is a true story, as I was telling you, near Michigan in US. Michigan is a state close next to us. I'm in Indiana, Michigan is the next state. So there was a lot of contamination, and people who were living in that area, they were getting cancer. But these were, simple people. They had no idea that the water was so contaminated, and that's why they were getting cancer. And somebody figured it out. So somebody fought a case for these patients. So obviously the people who were creating all this dirty water, they fought it, and there was a big fight. And then finally the lawyer who was fighting for that, right? That lawyer won the case, and these people were paid millions and millions of dollars. Okay, they still got cancer, but at least they were paid, right? So there is a very famous movie based on this story. Okay, so so what I'm saying is that today we will also talk about the environment, how much environment is controlling our diseases. Okay, so that is why lifestyle and environmental exposure, socio-economic status. You are rich, you have access to everything. Sometimes people are rich, but they're living in a place where they don't even have any knowledge about what to do or what not to do. So all of these things add up. Okay, so everything has to be considered to have the precise medication. So what are precision medicine disparities? Okay, so disparity in precision medicine means that not all patients have equal opportunity. This is different from what we are discussing here. So look at this sentence. Not all patients have equal opportunity to benefit, not receive, to benefit from advances in personalized medicine. If you replace this benefit word from receiving, then it is just a simple thing of rich versus poor. Somebody can receive it, somebody cannot afford it. It is not that. It is saying benefit. So benefit is a larger umbrella. Okay, even if these people can receive it, right, they may not benefit from it. Why? Let's look at that part here. So increasing diversity in research. So what happens? This will not make sense till we go to the next page. So disparity in precision medicine refers to unequal access. So rich people have access to it, poor people do not have access to it. That's one part. But this is very representation. What is representation? When they do clinical trial, right, in a country where, let's say, 50% people are white, and then I don't know, I'm just making up the numbers. 20% people are black, 20% people are brown skin like Asians, and then 10% people are Alaskan, Hawaiian, not Hawaiian, um, Native Americans. So very different people, right? Genetics wise, right? So when they do clinical trial, white are 50%, these people are 20%, 20% Asians, 10% others, right? But when they do clinical trial, 90% people are white, right? So the black people, Asian people, they are not represented in clinical trials, correct? So if you think even in precision medicine that Caucasian people, white people will have different kind of things than Asian. Then, like Chinese people, they have alcohol dehydrogenase deficiency, but we are Asians too, but we don't, right? So different people have different races have different kind of things. So if clinical trials, if our whole knowledge about cancer is only coming out from Caucasian people, if you use that criteria to some other thing, it may not work. So even these people are not taken equally in clinical trials. You don't have enough people of other ethnicities in clinical trial. That is also health disparity. So that is the meaning of unequal representation in clinical trials, right? I will tell you case studies about that. Okay, benefit that different groups of people experience in the development and delivery of precision medicine. So that is why even if Asians are rich, they are not going to get the right disease because they don't even know what is the right disease for an Asian person having this kind of cancer. Okay, so precision medicine, which aims to be perfect for one person, how it can be perfect for that one person because you don't even have enough knowledge about that ethnicity here, right? That is health disparity. Okay, so disparity exists between limited tested and minority and underserved groups, underrepresentation in clinical trials, cost and access barrier to molecular diagnostic. All of these things contribute to health disparity. So these patients miss opportunity for life-saving targeted therapy. We have it, we just don't know what to give to these people. Okay, so that is why, see the survival rate, you can see here. Same disease, same thing, survival rates are different. because of health disparity. Okay, so let's look at one all of these factors, the key aspects. So obviously I did not know, before I put this presentation together, I did not understand myself the extent of health disparity. All I understood before this presentation was that, few people get more aggressive cancer than other ethnicity, and that was my understanding of health disparity. Okay, so genomic underrepresentation. Most genomic and biomarker studies are heavily biased towards population of European ancestry, basically Caucasian people, right? Underrepresented groups include African, Hispanic, Latino, indigenous person. Indigenous person, like Native Americans, people who are basically really are in minority. Okay, Asian subpopulations are less included, which limits the applicability of finding. So biomarker based tests and drug responses may be less accurate for underrepresented populations such as these guys here. So lack of diversity in research creates disparity in who benefits from precision medicine. If a Caucasian gets, we can more accurately diagnose that disease, right? I will show you the case studies where Asians were given the wrong drug because they didn't have enough information. They were non-smokers and they were given these things. Access to technology and care. This is good. Rich guy has access to the health care. Poor guys cannot access it. It is there, but they cannot access it, right? So rural areas, low-income patients and underserved communities often face barriers. Cost of sequencing, limited insurance coverage, fragmented healthcare system can prevent access to proper health care. Okay, biological misrepresentation. Biomarkers validated in one population may not perform equally in others. So something which is validated in Caucasians may not work for African Americans, may not work for Asians, and vice versa. Okay. So this can lead to inaccurate diagnosis, risk assessment, treatment selection, everything. Then people who do not have access to these things, they are not even, sometimes, suppose they live in rural areas, right? So I live in Indiana, and Indiana is a, vast majority of it is farmland, but it also has two biggest universities there. So a huge population is highly educated because students are educated and faculty are educated. But the rural Indiana, people are not educated. They are not even high schoolers, right? So they don't know, they don't understand healthcare or anything like that. So, when COVID came, they had mistrust in vaccine. They thought vaccine is something which is supposed to be very bad. It does bad things to your body. So they were resisting vaccine with all their power. Okay. So that is what it means. Mistrust in healthcare and research among historically marginalized people. You know, vaccine can once in a while can have side effect, but it has also saved life. This is my view. It is the view of many other people, but then other people have other views too. So the point here is that if you can educate people, then they can make their own decisions. But if they are not even educated, then how can they make the right decisions? That's the point here. Okay, so they don't have health literacy, communication barrier. Then comes the environment. This is very important. Okay, so this picture tells you about the health environment. Okay, so here there is huge population like New Delhi. Okay, sorry, not population, pollution. Okay, huge pollution, right? Vector-borne diseases. Vector-borne diseases are those diseases which are, spread by mosquitoes and fleas and all these things, right? Like malaria, dengue fever, right? These are all vector-borne diseases. Racial disparities, okay? Health coverage, bad or good, okay? Disaster areas, right? Now, disaster areas is like that Michigan, the whole water was contaminated. Do you know what are food deserts? I did not know. I had to look it up. Food deserts are those places where you do not have access to healthy food. So the proper grocery stores are not there, healthy foods are not there. Okay? So people are eating really the wrong kind of food. And there are those areas where you don't even have access to healthy food. And then there are those areas where you never see trees or anything. You are living in a very polluted, no green areas, right? So they all these things are bad for us in the long run. So that is exposome. Our health is affected by, obviously, by the environment. Keep in mind, most of the famous diseases or the bad diseases like cancer, Alzheimer's, Parkinson, only 5% is genetic, 95% is the environment. Okay, so environment plays a very important role. Okay, so factors associated: social stressor, discrimination, poverty, violence, air pollution, lead, noise, occupational hazard, green space, absence of green space or presence of green space. Like IIT Indore, so much green, good for us. Living in a, I don't know where, but no green space is really bad. Concrete jungle, bad. Okay, healthcare access, personal care products, diet, exercise, substance abuse. Usually, there are pockets in Indiana, there are villages in Indiana where things are very, very rural. They don't have too many people like us go there, right? So they are very closed communities. And I have met people from there because some of the people still come out and study and go for higher education, and they said that 90% people in this area in my village are drug addict. So if you go around, people are just completely under the influence of drug. And this is true everywhere in the world. There are always these pockets where more and more people are abusing, so they have substance abuse problem. Okay, so so these, obviously the environment affects the epigenome, it affects the whole physiology of human this thing. Okay, so let's talk about exosome. So nobody who manages this class is here, right? Nobody except us. So we can keep going. Hopefully they'll remember us to remind us when to stop. Okay, so all the people who are controlling are out of the room. So it's our show. So we keep on going till they stop us. Okay, exposome. Okay, so this is the environment thing. Okay, so the person's physical, cultural, social environment plays critical role in their health. If you're a happy person, if you're in a happy place, your body is happy, your stress is low. If you're in a stressful situation, your environment is stressful, it will affect your body tremendously. Okay, so environment, home environment, huge importance. Education, employment, eating activities, drugs, everything. Suicide tendency of suicide, depression, all of these things, they are not just an attempt to document the story of a person's life, but to truly understand everything that embodies their reason that might have contributed to their disease. People who are angry all the time, real angry, they tend to have more liver cancer. Okay, anger is pretty bad because it really changes your physiology, and that contributes to cancer. Too much hatred towards things also leads to cancer. So what these emotions are creating real changes in our body that is fostering the growth of cancer. Okay, so so scientists lack reliable ways to measure precisely how non-genetic factors can help health, and must develop ways to measure the effect of the social, physical, and cultural environment. I will give you some cases here. Okay, so one of the best cases, that there is a very famous person at Harvard. He's of African American background. So people who live in South Chicago. So South, Chicago, you have all heard. So usually on the southern part of South Chicago, people who live are relatively uh, financially less abundant people. They are of African American origins. They don't have, they are not very educated, and there is a huge crime rate, right? And they also have very high blood pressure. So for the longest time, people used to think, oh, they have originated from Africa, and this high blood pressure or hypertension and all these things are in their genetics for the longest period of time. But if you go back to Africa from where they came, those people do not have health problems at all. They don't have hypertension. So why these descendants have hypertension, right? Because they are marginalized. They don't have access to education that much. They don't even know much about education. They are living next to highways, industrial waste, pollution, because those are cheaper areas, right? They are not green areas. So these factors are the one which are giving to hypertension. But up until now, we were saying, well, they got it from their own parents, right? So that is not true. It is the environment which has given that. They did not inherit it from their parents, right? So that is exposome, your social, your own world in which you live, your friends, your family, your environment, your social life, your professional life, that contributes to your well-being. Okay, so where you live, work, play, that's what is. So genome obviously plays a role, genetics plays a role. The complete set, genome is complete set of individual DNAs, which is the biological blueprint. If someone is BRCA1, BRCA2 deficient like Angelina Jolie, definitely more prone to developing cancer. So their genetics is playing a role. Okay, sickle cell anemia, genetics is playing a role. But exposome is totality of environmental exposure, physical, chemical, biological, social, lively. This determines how genes are expressed and how disease risk. So suppose you are smoking a lot or someone is smoking a lot, right? They can change their epigenetics so that the wrong things are expressed, good things are not expressed. Epigenetics plays a very important role because it can suppress good genes, it can activate bad genes. Okay, so air pollution, increased risk of asthma, cardiovascular disease, smoking alcohol, cancer, liver disease, stress, diet, infection, chronic diseases. Okay, so if this totally depends on your environment. For every disease, the biggest contributor is the environment. Environment also includes our own habits, our own diet, our own, whether we are smokers, whether we drink alcohol, whether we eat chips all the time. Everything adds up. That is all environmental. Okay, so genome and environment together, they determine the health thing. Sometimes genetics becomes very powerful. So for example, I know families where everyone will develop diabetes, whether you eat sweet or not sweet, but you get those genes from your parents. So they are very prone to diabetes. So you have to be very good. People get obesity genes. Parents are obese, children are obese, right? But you can always fight it off. You are more prone to gain weight, but if you exercise, you will be thin. You have to work harder than other people who have slim genes, right? But so epigenetics, this goes without saying, epigenetics plays extremely important role, and exposome actually controls epigenetics. Okay, so the take home message is that genome does not say that you will have diabetes. It will say you have high potential to get diabetes. You have high potential to get this disease. It is not giving you the final verdict. It is just telling you potential. Okay, and exposome says, okay, you live in this environment, I'll make sure you get this disease, or I'll make sure you don't get this disease. Okay, that is that. So it is the dynamic interplay between the two. Let's look at the case studies. You will really enjoy because these are real cases and you will see how it goes. Okay, so this is that. African Americans traditionally are thought to have high blood pressure because they thought it is a racial trait. These guys have in their genetics. However, average blood pressure from West Africa, from where most of the African Americans trace 80% of their genetic roots, to the West Indies and then to Chicago, you will see a stepwise increase in blood pressure. So in West Africa, they don't have hypertension. The closer they come to US and come to Chicago, blood pressure keeps shooting up. Okay, so this is not a genetic tendency, and it just happens people who live in the south side of Chicago. By the way, Michelle Obama, President Obama's wife, she always tells she's south from the south side. That is, that is the poorest place, extremely high criminal thing. You don't want to go to south side because it has such criminal thing. So she always says a small girl from south side of Chicago went to become the first lady of United States. And first lady of United States, you can say, well, her husband was the president, but you know what? She's extremely educated lady. She was a Harvard graduate. She became a Harvard student on her own, not her husband's thing. So she's extremely accomplished lady. So her genetics were fine. Her environment was as bad as it gets, right? And she rose to become what she is today, right? So your environment can be adverse, but you can rise and come out of that environment. You don't have to stay in that environment. You work hard, you get out of that system, right? She always says, White House was built by the slaves at that time from Africa, right? And she said, when she went to the White House, she said, every day I walk around and I said, my ancestors built this house, and today I have the privilege of living in this house. So she was very mindful of that, and that is very motivating for people who are stuck in bad situations in south side of Chicago or similar areas, that they can get out and they can rise to the highest level, right? So even if the environment is bad, you can always get out of that environment. Nobody is stopping you from that, right? Case study 2, breast cancer outcomes. So African Americans, whenever they get breast cancer, it is always poor prognosis. So African American women have higher mortality from breast cancer compared to white women despite similar incidence. Why? Because they don't have access to mammography. By the time breast cancer is diagnosed, it has truly advanced. White people, no symptom, you can pick it up, you can quickly treat it. They also get more aggressive breast type like triple negative breast cancer, socio-economic barriers to early treatment. Very good example of health disparity. Then diabetes in Native American communities. So Native American population have some of the highest rates of type 2 diabetes. Why? They have limited access to healthy food, right? So what happens is that the story of Native Americans. So Native Americans are these are Native Indians also. Like, when Columbus said red Indians, these are the people, right? So he was looking for India, he reached America, and they were very fair, so he called them red Indians, right? So these were the original people who were living there, and then people of European descent came, then they became the Friends, then they owned in the US, and these people were pushed to some small areas. So they are pretty isolated people, right? So those Native American peoples, they have limited access to healthy food. They have faced historical trauma. They were really marginalized, right? So systemic marginalization, inadequate access to preventive health care, which all other people have. So because of that, it has resulted in genetic predisposition towards diabetes. If you don't eat healthy food, diabetes is very easy to develop. Okay, it's a very easy thing. Okay, COVID-19 pandemic and racial ethnic minorities in US. Black, Hispanic, Latino, indigenous population faced disproportionate infection, hospitalization, and death rates. Not surprising, you can predict now, even during COVID. Higher likelihood of being essential workers, those people who really help. So, COVID time was so bad that people were not even allowed to see their loved ones if they were infected with COVID, right? They were taken. So, but there are people who are taking care of all of these patients and everything. So they were exposed, right? So these are the essential workers. Crowded housing, less access to testing, vaccination, pre-existing conditions like diabetes, hypertension, of course, higher fatality. Okay. Physical environmental, higher exposure in low income urban minority neighborhoods, more asthma, more cardiovascular diseases. I have heard that somebody was telling me that in Delhi, if you walk for even half a mile, you get you just get completely out of breath. But if you walk in a nice area, you can walk for miles and you don't get it because of the air pollution, right? So imagine you are living in that area day after day after day for it will help that. So housing condition over, so this is that Michigan led crisis. Contamination disproportionately affects marginalized communities. Okay, poor ventilation, mold in the house, infection, respiratory illness. Okay, now this is food desert, lack of access affordable healthy food leads to obesity, diabetes. We were talking that some people are not educated enough to buy healthy food, not that they are poor to buy healthy food. So so many times we see that you go to a supermarket and people have carts full of stuff, and if you look at their cart, we also buy stuff, but our thing is in the bottom because we are buying vegetables and everything, right? So that's their raw vegetables and fruits and things like that, but their cart is full up to that because there are no vegetables, they're all tin food. So overall their food is not much than ours, but ours is raw here, but these are all preserved, right, in boxes, in tin things, right? So they will save you time. You open the tin, you heat it up, you this thing. But all of them have preservatives, and that is very bad for you. It is as bad as it gets to buy tin food and eat, right? So people, either they don't have time to cook food, either they don't understand what is healthy food. Oh, sorry. I thought somebody told us finally you can stop. So I was looking very hopefully. So yeah, but they are not educated enough to know that they should not eat this kind of food. And every time I see, I feel like, obviously I have to keep my mouth shut. Okay, but the thing is that they just don't know that they should not eat this kind of food, right? So, so food desert, green spaces, lack of opportunities for physical activities. At least nowadays, when they build big apartment complexes, they have some facilities, physical facilities. But the thing is that those facilities are so dirty because people are sweating when they are running. So if you run in those facilities, you are probably likely to get more diseases, right? Because they don't clean it enough. So basically, there are many factors contributing to diseases. So healthcare infrastructure, fewer hospitals, clinics in rural or underserved area, access to preventive emergency care is very poor. Okay, so this, okay, neighborhood safety, unsafe areas limit outdoor activity. So in many times in India also, we don't go outside after this thing. We know if we walk around in this area, it is not safe, right? So, so leading to chronic disease risk, education, lower education, unstable jobs, less health literacy. This less health literacy really came out during COVID time because there was a large number of people who were really, really against vaccination. Like there was this real big fight, no vaccination, no vaccination, no vaccination. We were craving vaccination, right? Like the fast, and then COVID came under control due to vaccination, right? Because otherwise it was just spreading and spreading and spreading, right? So, and it is not their fault. They just don't even know what is vaccination. Somebody was really saying it is a devil. You are putting a devil in your bloodstream because somebody told them that, right? So chronic stress from systematic inequities. This is also a very sad thing. So, we cannot say that all Asians are bad or all white people are bad or all black people are bad. They're good or bad everywhere in everything, right? But these people, these people will get labeled as bad all the time. So even if they are extremely good people, they are likely to be targeted by the police, right? If they see this person, okay, this person is this, obviously he has to be bad or she has to be bad, right? So Serena Williams. We all know Serena Williams, the tennis player, right? She said, I feel so bad if my nephew goes out. He has 10 times higher chance of being caught by the police than any other person there, right? So that is a reality, right? And that gives stress to the person, constant stress about the safety of that person, right? So all of these contribute, right? Okay, environmental health disparity, air pollution and respiratory, this thing. See, in many African cities, residents are exposed to higher levels of air pollution from traffic, industrial emissions, and biomass fuel usage. Children face higher rate of asthma and respiratory infections. Okay, so environmental regulation, there is no environmental regulation, lower access to healthcare for diagnosis treatment, right? Rural reliance on biomass fuel for daily living. So all of these things, right? Now we are saying greener environment, right? We should have cleaner this thing. We are saying, I mean, there are obviously huge amount of pollution all over the world, right? There is global heating going on, but everything just this one, lack of Medicaid expansion, inadequate insurance coverage, barriers to care. People of color, people who are darker in color, people of color often are located near toxic waste sites or industrial pollution sites. When you go to US, right? So you will see like really beautiful houses, and you will see like one-story houses like so, and you could always see that the houses which are next to highways, which are next to the road, they are just really poor looking houses. The houses which are further away from highways, they are big and huge and beautiful houses. So just by location, I have never ever seen a beautiful house next to a road, like main road, because that area is very cheap if it is next to main road because there is constant traffic. If it is next to the industry, higher crime rate because the houses are cheap. So all those other people, they live in other parts of the thing. So living next to a industry which is releasing a lot of waste in the air, those areas are a lot cheaper than if you go to places where it is much cleaner air. Okay. Chicago has stark neighborhood level disparities in environmental exposure. Black and Latino neighborhoods are disproportionately located near highways, industrial zones, and waste facilities. Okay. Asthma hospitalization rates for black children in Chicago are 5 to 6 times higher than white children. These are real numbers. Okay. Heat waves like 1995 Chicago heat wave caused disproportionate deaths in poor segregated neighborhoods lacking green space and conditioning. Okay. Then lung cancer in African American person, historical lower rates of EGFR testing. So this is what happens is that uh, there is a story, a black woman went with lung cancer. They would not even do EGFR testing for her, and they treated her with chemotherapy because it was too much effort to do EGFR, and she had EGFR based lung cancer, breast cancer. So she died because she was given chemotherapy. Okay, so they don't even test for which cancer it is, just give chemotherapy or radiation therapy. And okay, so take home message is that precision medicine without equity is incomplete. Okay, you can develop all the precision medicine. You are not even taking all the diverse population in your clinical trials. You have to know that, right? And keep in mind, the more population you will include, the more knowledge you are going to get. It is not about that person. It is all about your own knowledge in how disease is expressing in different ways in different human beings, right? So you can talk all about biomarkers, you can talk about precision medicine, but you have to have all the representation there. Then you have to make sure that all of them get equal access, right? I think this is the last one. Summary, the environment is not just physical, it includes social structure and policy dimensions, right? So this is a very important part of this one. So we hope in 2030, which is really next door now, okay, we will have more diversity and inclusion, we will have big data. So the best part is that, and this will come towards the very end of this course, right, when AI comes, AI shows the face here. But the thing is that there is so much data, but they are all segregated data. Like people here have their own data, people here have their own data. So we have to combine all the data, right? And we cannot handle it as humans. So the AI can handle all the data pool from all over the world, right? And then we have to integrate all of those data. So the last lecture will tell you that human mind cannot comprehend this amount of data at once, right? Because we cannot include every kind of thing. That's not even possible. So that's where AI can come as a collaborator in sieving through all this data and then really helping us. But we have to keep the final decision in our hand, not let AI make the decision. Okay, we can integrate, we can confirm, we can do that, and then we decide, okay, this is the precision medicine best suited for that. Okay, all right, this is self-explanatory, all the tests and everything. All right, nobody came for us. We went directly for 2 courses. It's 12. No, the lunch. No, lunch is at 1. Oh my God, we can even go. All our hosts are this. No, don't call them. We can just have some fun time. उसको बंद कर दीजिए, हम लोग थोड़ा relax कर लें. ऐसा करते हैं ना, we can do this way. It's up to you. They are not going to serve us lunch until 1 because the lunch is probably not even ready. So we can just take 5 minutes break and then finish the 4th lecture. Then we have lunch. Then we just we'll finish up everything by 3 o'clock. Better, right? Yeah. I have too much adrenaline pumping, so I can go, no problem. I don't even need 5 minutes break. है ना? तो पर हम लोग 5 मिनट्स ब्रेक लेते हैं सब लोग, क्योंकि आप लोग को एब्जॉर्ब भी तो करना पड़ा, मेरा बोलने से तो खाली नहीं चलेगा, है ना? तो 5-10 ऐसा करते हैं ना, 10 मिनट में विल रिज्यूम. सो बाय लंच विल फिनिश द 4th one and then we can have fun time during what is the last part, tutorial. Yeah, I was told कि tutorial में I am supposed to ask you questions, but I want you guys to ask me questions because I will keep asking questions as we go along anyway. So it is 11:56. So let's start 12:05.
There's one thing, while you're keeping them, right, they die on their own. After some time, they die. Cell line. First question, what made two different, various cell lines coming from the same cancer? Why there is different cell lines? Is it because some other gene got mutated? That's why they cured.
So give me a specific example. Are you saying breast cancer has many different cell lines? Yeah, these are different kinds of breast cancer.
Just
So
because of the upregulation of certain genes in different cell lines.
yes, they are completely different. Yeah.
That's why there are different cell lines.
Some are aggressive, some are not aggressive, some are pretty benign. Some will form a huge tumor, some will not. I'm okay. Some will form a huge tumor, others will not form a huge tumor. Some will respond to this therapy, others will respond to this therapy. Some express this, some do not express this. So they are all breast cancer, but they're just totally different from each other.
Reason being is
Different proteins are expressed. Yeah.
okay. So I have noticed, like we all study while we dealing with cancer, is they die on their own after some time, but we don't treat them. We treat them for therapy, right? So they die on their own.
But you are treating them with drug, right?
That's fine. If we don't take care of them, they die. If even the temperature is not favorable, if it is incubated,
Yeah, because when you don't change the media, so they are contact inhibited. So if they get too close to each other, they cannot divide anymore. Then there is nutrient deprivation. Then the medium become acidic. So all of these things is too toxic for them. So you have to keep them happy at a lower confluency.
can you take this point to the treatment? So
No, so here is the thing. You are asking a good question, but the cells that you use in the lab, human cells are different when they are in the body. So in the lab, these are immortalized artificially. So they are good for us to do some kind of work there, but that is not how cancer actually develops in the body. So suppose you are working with MDMB231 cells, right? So they divide every 24 hours. But the same cancer of from which MDMB231 originated, in human cells it is dividing every 10 days. Okay, in human body these are not treat, so they have been immortalized by different genes so that they can stay in tissue culture infinitely. But that is not the case in human body. If we take the same cells out of the human body, they will die within 10 days or 5 days, because these are not immortalized. So they will become senescent and they will eventually die, all of them. So keep them growing for generations and generations, our generations, right? Like for decades and hundreds, 100 years, or not 100 years, but 70, 80 years, some cells, or maybe, yeah, 70, 80 years. So for going for that long, we have made them immortal, and they divide much faster than they are dividing in actual human body. Okay, so they are different there. So so if they are dying there, that is due to nutrient deprivation and things like that. So how are you going to, so when you are giving radiation therapy and things like that, you are killing them, right? You are killing them by damaging their DNA, and you are giving them PARP inhibitors so that they cannot do that. But how can you selectively deprive them?
Looking at the perspective, they die on their own. Since my doubt got clear now, but earlier I had a doubt कि if you tend to keep them stationary and they will die, like it's not proliferating. If we tend to keep an environment or create such type of situation which is not proliferative for the human, at that time we can give birth or
So let me stop you here before you complete it. So remember, in tissue culture, you are growing cells in 2 dimensions, but in humans, it is growing in 3 dimensions, right? So the same condition, what is happening in tissue culture, will not mimic in human because they have many different directions to go, right? But in tissue culture, touching each other is enough to close the contact, like kill them, right? If they become too confluent, they die because they cannot grow. So that part you cannot mimic in humans because they're growing in every direction. But now keep going on.
the solution. I have multiple questions, so I think they got mixed up right now.
That's okay, because even if you say tell me half the part, I will understand what you're trying to say. Go ahead.
I need some coffee if I'd left to drink.
No, 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 nothing. Thank you, thank you. Yeah.
So there is a cell cycle when life dies after some time. Can it happen in body also? If we make it at if it make the metabolically quicker, but
Quiescent.
yeah, but persistent. हाँ, the question was कि अगर सिर्फ long duration में hold करने से they can die after some time, then why can't we use it in medicine? Let them
So,
develop for long.
so tell me exactly what you want to do. Maybe we can do. Tell me. So, if there is a tumor growing, what will you do?
I'm supposing just like the half-life of a and our disease for example disease, is there any chance tumor have their half-lives also after that they die themselves?
No, it is not the case there. So what happens is that tumor, some cells die for sure. So this is a well-known fact. If the tumor grows very, very, very big, you often see some cell death in the middle, necrosis, like too much. You see just a necrosis there. So there are tumor cells which are dying, but not as much as normal cells. Okay, so many times, so here is the thing, when you are constantly mutating, like if you are a risk taker, in life and you're taking many risks, or even let's say you are investing money and taking huge risks, sometimes you will gain money, sometimes you will lose money. So the kind of mutations that are going on in cancer cells, many times they die because those mutations are not favorable. But even if a very small percentage of cells survive better than their parents because of the mutation, they take over the whole thing. So they are much more aggressive, they grow faster, they do everything, right? And they will take over. So what happens is that, so to mimic the condition that you are saying, people thought angiogenesis inhibitors. So you are giving them angiogenesis inhibitors, so you're cutting them their food supply, you're cutting their oxygen. So you would think that is more than enough to kill the cells completely. So now under hypoxic conditions, the new survivors come and they are so aggressive, you can keep on using therapy. So sometimes people say don't give angiogenesis inhibitors because the guys which will come out of that are even worse than the parental cells. But then you have to give angiogenesis inhibitors anyway, because otherwise the tumor is growing too fast. It just gains you some time. So the problem in the body is this: every time you give a therapy, they come out even more stronger. Something new. It's a constantly evolving disease. Okay, let's start now. It's 12:05. Okay. This one is an easier lecture because it has a lot of things that we have already covered today. So we can definitely finish it by 1 o'clock now. We really need some computer person here who knows how to operate this system, because this is frozen here. I can't even get rid of my PowerPoint and open up a new PowerPoint. Okay, let me go to the task manager and close everything that way. Okay, I'm closing PowerPoint there, so hopefully. So now we know what are biomarkers, and now we know where is my okay. Yeah, so now we know what are biomarkers and we know what is health disparity, right? Now we combine the two things, that how we can find biomarkers for precision therapy, which includes all people, right? More diversity. Okay. So for example, these are different kinds of patients and they have different biomarkers, and if you can identify the correct biomarkers, you're going to treat them completely differently based on their biomarkers. Okay, so remember I told you about 4 things. So we have already covered this and we have covered this topic in details. Now we'll look at these two more in details. So I will briefly tell you about biomarkers again, because this lecture was supposed to be on different days. Now we are combining because of that. So I will go over these biomarkers very quickly, and then we'll look at some actionable biomarkers. So now you have very clear idea how we can exploit biomarkers for precision therapy. Without biomarkers, there is no precision therapy, because biomarkers are the one which tells us exactly what we need to use, how much we should use. So prognostic biomarker, it tells us what is the outcome of that disease, whether it is bad, whether it is good. Diagnostic will tell you which kind of cancer it is. Predictive will say what is the best therapy for this kind of cancer, right? Pharmacodynamics will tell us what is the best dose. If we are giving HDAC inhibitors, are acetylation levels going up? Is it hitting the target? Recurrence, it will also tell us whether this cancer is going to come back or whether it is not going to come back. So I already showed you diagnostic biomarker. For example, BRCA1 mutation, if it is BRCA1 positive, if KRAS is mutated, if P53 is deleted, right? These are all diagnostic biomarkers. Okay, prognostic biomarker, there is a new thing I have included here. So for breast cancer, I was working with a pathologist once. His name is Sunil Badwe. He's at IU Medical School. So he works on breast cancer. So he's mainly a pathologist. And pathologist, as you know, he works with cancer patient. He does biopsy. And when he gets biopsy, then he uses it for his own research to see what is wrong with this biopsy. So he was the one who introduced me to this thing. So there is a test, like a kit test, which is called Oncotype DX breast cancer recurrence breast cancer thing. So what they have done, they have seen in breast cancer these 21 genes are upregulated or downregulated. So they have made a kit to check these 21 proteins. So how we came across together that I was working on Aurora E kinase, which is highly expressed in every human cancer, including breast cancer. And he was working on breast cancer. So I needed access to tissues of breast cancer. So remember, cells are okay, but tissues are the real deal, right? You have to. So that is how we set up collaboration. And he said, you know, your Aurora A kinase is one of the gene which is actually tested in Oncotype DX model. So imagine out of the thousands of genes, if 21 are selected and your gene of interest is there, obviously it is very important gene, right? So Oncotype DX breast gene test for many different things, including HER2, Aurora A kinase also. And then they identify those patients who may benefit for Herceptin, for chemotherapy, immunotherapy, things like that. Okay, so it depends the magnitude of chemotherapy benefit. It's supposed treatment decisions. So prognostic marker for Oncotype DX breast, a small sample of breast tumor tissue is collected and analyzed. So my friend was actually analyzing it, right? And so they measured the activity of 20 genes that are associated with cancer recurrence. Okay, a recurrence score is calculated based on gene expression thing. So if it is higher recurrence score, bad news, lower, good news. So look at this thing here. So low recurrence score 0 to 25 indicates a low risk of recurrence, and chemotherapy is unlikely to be beneficial. So why chemotherapy is unlikely to be beneficial? So chemotherapy is very toxic. Okay, so if suppose somebody has localized cancer, you will not give chemotherapy. If it is localized, you would probably like to do surgery and remove it completely. And if you have to give chemotherapy, just give it to some extent, like even if their subcells escaped, they should be killed off, right? But you will not give chemotherapy. But if the tumor has spread everywhere, now you cannot do therapy everywhere. You cannot do surgery everywhere. The person is not going to survive, right? If it has gone to lymph nodes, now you have to give systemic therapy so that every cell in the body is exposed to the drug. Now you will give chemotherapy, correct? Because now it is literally a question of life and death, right? So when it is low recurrence, so you would like to do less aggressive therapy, clean it up, clean up the mess if it is localized. So that is why chemotherapy is unlikely to be used. Intermediate recurrence to chemotherapy may be considered based on other factors. Really high, now you know you have to use chemotherapy for this patient, right? So by this one, this is just one simple thing. But suppose it is Aurora A kinase is overexpressed. Now you will give Aurora A inhibitors, right? This is also, Aurora inhibitors are not FDA approved as yet, despite this being a very, very important target, because they are very toxic, because Aurora is expressed in every cell of our body. Okay, so patients cannot handle Aurora inhibition in normal cells. Okay, so benefit is that it personalized treatment decision based on individual risk, whether we should give chemotherapy, whether we should give HER2 inhibitor. It tells you exactly. Limitation is that not suitable for all breast cancer patients, triple negative or HER2 positive cancer. So for example, there are many different kinds of breast cancer. So 21 genes is not an adequate representation to look at all the pictures there. So this is limited information. Still 21 genes is providing you limited information. Okay. So does not predict the risk of distance metastasis. This 21 genes will not tell you whether this is going to metastasize or not, just because it does not have enough representation. Okay. So I'm quickly reminding you about the biomarker. Since we studied just 1 hour ago, you already know the diagnostic, the prognostic, the predictive, pharmacodynamic. So I'm just going to go no response, right? Predictive biomarkers will predict whether a patient will respond. EGFR mutations, EGFR inhibitors, KRAS. So KRAS, if it is wild type, it predicts response to anti-EGFR because they talk to each other. And that is how you can predict different kinds of things. Okay, so you already know this is telling you survival. See, this is less survival, this is longer survival, right? So that is prognostic. Predictive biomarkers, if you have PD-L1, you know immunotherapy will work, right? Because PD-L1 will inhibit PD-1 and T cells will sleep, so you have to use immunotherapy, okay? So this is again prognostic predictive biomarkers. And so this oncotype breast cancer, similarly there are other kind of tests which will test. You also saw case studies where in this woman they have tested every different things. KRAS is wild type, ALK is negative, this is negative, right? So pharmacodynamic is the one which will tell us if you are using HDAC inhibitor, are acetylation levels increasing? If you're giving anti-cancer drugs, are Ki-67 levels decreasing, right? These are all pharmacodynamic. You are actually seeing if the drug is doing what it is supposed to do. If you give Gleevec, PCR levels should decrease. That's all pharmacodynamic biomarker. Okay, this is the figure I wanted to show you since morning. Okay, so this is there. So this is pharmacodynamic study here. So this is the baseline level of one marker. Okay, now you started the treatment. See how fast it decreased here. And then it slowly increased here. And now it is resistant. So pharmacodynamic biomarker is measuring, this is really good news. And slowly it is developing resistance and it is still increasing, right? Therapeutic biomarker act as a direct target of the therapy. So BCR-ABL is the target of imatinib, right, in CML, PD-L1. So if PD-L1 is expressed, we will use immunotherapy targeted towards PD-L1. Okay. Risk biomarkers are just predicting the risk. You already know that, right? BRCA1, BRCA2 is a good one. Safety biomarker. This is something I have not told you, but we will discuss this in details tomorrow. Okay. What are safety biomarkers? So, like I was telling you since morning, 10 people have identical identical disease, exactly the same stage, but we all will respond differently. Why we will respond differently? Because this is where the genetics come into play. Okay, so depending on our genetics, we may all respond completely differently to the same drug for the same disease. Okay, so you know hydrocodone. Have you heard of hydrocodone? Anyone has heard? I think hydrocodone or codeine, something like that. It is probably codeine. So when you have some surgery and you have really huge amount of pain in your body, codeine, right? Codeine, yeah. So like if you have dental surgery or any other surgery or any accident and you have huge amount of pain, people are given codeine, okay, to control that thing, and people get addicted to that codeine and they can never get out of it, and that is what leads to drug abuse. Okay, and that is a very serious reason. I know many people, I mean, I don't know any of them personally, but I know their friends or family personally. So somebody met with an accident and the hospital doctors give them these kind of drugs like morphine or something, and then they got addicted, and that time the doctor was giving them because they could not survive with that pain, and then they got addicted, and now they cannot live without that addiction, right? So uh, but at the same time you give codeine to other people, they will not get addicted, and every time you give them codeine they are going to throw up. So other people are enjoying codeine and these people are throwing up codeine because it is just really toxic to them. So they have the same disease but they are reacting very differently, right? That is because of our genetics. Okay, and we will discuss that in great details tomorrow. That comes under pharmacogenomics thing. So if we incorporate pharmacogenomics there, then we can predict exactly. So suppose we do our genome thing, like they don't check the whole genome, they check maybe 20 things there. And those 20 things will tell them exactly, should I give this drug to this patient? And they will know, no, this person cannot use this drug and this person is going to feel worse about that, right? So that is what is they say, that if this person has this mutation, this drug is for HIV, this person cannot tolerate this drug. So if somebody has HIV, they do a quick genetic test, PCR, 1 day test, and they will know you cannot give this drug if this person has this mutation there. If they don't have this mutation, this drug will be phenomenal for them. Okay, for this one, you cannot give it to some patient who cannot tolerate this disease. This is just due to mutation in subgene. Okay, tomorrow I will give you that. So this is also, that is risk biomarker, which I did not tell you before. So now we know about all the biomarkers, we know about precision therapy. So let's go and really look at some cases here. So now we will look at eliminating precision medicine disparities by identifying actionable biomarkers. Now we are combining the two things, disparity in health research and finding the biomarkers for that, so that we can find the exact thing. So role of actionable biomarkers. Actionable biomarkers means molecular or protein markers like DNA, RNA, metabolite, whatever it is that guide specific treatment decision. EGFR mutation means you have to use EGFR inhibitors for lung cancer. HER2, Herceptin. BRCA1, PARP inhibitors. Identifying them levels the playing field, ensures patient get the right drug for the right patient at the right time. Okay, so how do you identify actionable biomarkers to reduce disparities? Early testing, universal biomarker screening and diagnosis prevent missed opportunities. Okay, population inclusive panel. It is very important that we have the complete picture for all across the board, all kinds of patients. Okay, so for example, it is possible that people of Asian origin, when they, maybe we don't even know as of today, that people from India versus China versus some other Asian country, we have different kinds of mutation, right, in some gene, and we may respond differently because of those mutations. But if we know in advance, then we know exactly what to give. Okay, so population inclusive panels, designing biomarker assisted validated across diverse ancestries. Liquid biopsy, that is the best thing. You just take out the blood and you figure out what the disease is. You know, we had a faculty member in our department. He has now retired. He's a very famous chemist. His name is Fred Rainier. So he was a mass spectroscopist, right? Analytical chemist, world famous analytical chemist. But he was very interested in biology. So he was studying glycosylation pattern. So glycosylation pattern means proteins are decorated with sugar and they form all different things. So he found, and that is about 20, 30 years ago. So that was extremely novel at that time. He was one of the first person to find that glycosylation pattern is different between normal and cancer patients. So obviously it is not easy to get access to cancer patients. So he used to work with dogs with cancer. So normal dogs, dogs with cancer. And then he took their blood and he was a mass spectroscopist, right? He's the one of the person who developed all these huge techniques for mass spectroscopy, right? So extremely bright guy. So he showed, and he was telling me, by the way, he's set up my LinkedIn page. I never wanted to come to LinkedIn, and he said, you have to come. You have to come. This was, I don't know, 15 years ago. Since that day, I've never been there. But he was the one who created my LinkedIn page, and I'm still what he created that. So it has never been updated since that date. And I cannot delete it. Once it is there forever. You cannot delete it. Okay. But he was my friend. So he was telling me, you create like this big chip something, and I don't know how it works, but you take the blood from normal versus the diseased dog, and by looking at the glycosylation pattern, the sugars that are shed in the blood, you can predict cancer even before it develops in the dog. Okay, so it was really, really powerful. So if you can develop non-invasive thing, people are more likely to get it tested. If you say they have to do a surgery to get the biopsy out, they are not likely to do it, right? And of course, education and awareness about these diseases is very important. Training clinicians and empowering patients in underserved communities. Okay, so improve diagnostic across population, personalized treatment access to all, reducing trial exclusion bias. Cost effective is very important, especially if insurance companies not there, or if by the way, healthcare is best in Europe, not in US. In European countries, healthcare is absolutely the best. You can get extremely good healthcare, equal to all. Okay, and monitoring and following up is very important. Okay, so this is all the same things that I have told you today. They are all listed here. So I'm not going to go through this, but you can read it here. We have covered all these topics in detail today. Okay, we have to have standardized care guidelines, improve treatment selection, enhance drug discovery. This thing, the use of biomarkers have shown to increase the success rate of clinical trials. Okay, accelerating the develop and FDA approval of new targeted therapies. Okay, it is very important for community engagement and awareness. So these days, I'm sure it is here too. There are many funding agencies. They will give you money if you have some component to raise awareness, to engage community, to educate them about diseases. Okay, so this is very good. Let's look at case studies. That is where it gets most interesting. So lung cancer in African American patients, historically low rate of EGFR testing compared to white patients. So this is the story I was telling you, that this black woman had lung cancer, or I think she had breast cancer. So instead of checking for HER2 or EGFR, HER2, EGFR, they belong to the same family, by the way, right? So instead of checking that, they just gave her chemotherapy and she died. And if they would have given her the right drug, she would have survived. Okay. Case study 2, EGFR mutation in lung cancer. This is interesting. Never smokers Asian population. Now, Asian population is misrepresented in clinical trials, so they don't even know. So disparity, lung cancer in Asian and never smoking patients was under-recognized. So chemotherapy was given because they didn't even know. These guys have never smoked, nothing. They gave they gave chemotherapy. These people, they died eventually. Okay, but the actual intervention would have been EGFR testing because it is due to EGFR, and they never tested EGFR, so they just gave them chemotherapy. Okay, also it is true that um, many times even the hospitals are there, even they have access, they are not willing to test it for you. Okay, they will just give you chemotherapy unless you put some kind of pressure. No, you have to test it. This happened to someone I know extremely well recently. Okay, this is a gone case. No, we are not going to even worry about it. And then the son was so good, he said, no, you have to. So he got a lot of people involved, and basically they had to change the whole regimen, and this person survived. Okay, so sometimes you have to also push it to go in the right direction, especially if you have knowledge. And the funny thing was that the son is a IT person, so he has zero knowledge of biochemistry. So he used to talk to me all the time. So I was the person who was guiding him, and the second person who was guiding him more than me was ChatGPT. So he studied everything in ChatGPT, and then he would call me, Didi, I read this and this and this, and we would discuss it. I said, use ChatGPT, but ChatGPT can mislead you too. But he would just put everything in ChatGPT, and then he would talk to me. And I really am so proud of this guy. He just saved his father because he was so adamant. Like he took his he's in US, his sister is here. The sister took the father to Max Hospital in Delhi, and they would not look at him. And he lives in Kanpur, and they took him to Delhi. They would not look at him. This is a gone case, this and this. And this guy, the son, he talked to people in Seattle, he talked to doctors in Chicago. Chat GPT was guiding him all the way, what to do, what not to do, and everything. And he paid them. He paid these guys, got all the reports. He did all the recording. He was sending me the recording also, that so that I can hear everything in the original format. And then he was talking to them like everything. Can we do this? Can we do this? What is the option? This doctor said there is 1% chance. And the first thing this guy said, which brought tear in my eyes, oh, there is 1% chance. Oh my God. I thought I cannot take because it was just too much for me to take that. But the bottom line is that this person survived, because now the doctors in Max Hospital said, oh, these people's top oncologists from US are involved. Now they are paying attention. And before that, they were shouting at this guy's sister, you know. So sometimes you have to push the system to get the right therapy. It is out there. If somebody says it is not going to work, it doesn't mean it is not going to work, you know. So, all right, let's go to case study 3. BCR-ABL. BCR-ABL, we know drug is out there. So if you identify BCR-ABL, so disparity, because it is a leukemia disease, right? Chronic myeloid leukemia. So you have to do bone marrow transplant. But if it is inaccessible, obviously it is a bad thing. But at least intervention is very simple. Simple PCR testing. BCR-ABL will come in a day. In a lab, you can know in 2-3 hours if that is the problem. You make a primer towards BCR thing and ABL where they are joined together. If it is positive, you know it's BCR-ABL, right? And off the shelf drug. I mean, not commercially available, obviously. The oncologist has to prescribe it. But this is very important to do this kind of testing. So strong, actionable biomarker, simple assay, equity. Anyone can access it. It is not expensive. Gleevec is expensive, but okay, the testing is not expensive. And I'm sure Gleevec will become generic soon. Then it will be very cheap also, right? HER2 in breast cancer, you already know Herceptin, right? Okay. This is another case. So I briefly told you about this. This is a HIV medication. So it is a very effective medication, but you cannot give it to every person. You cannot give it to every person. Few people are going to react so badly, and I will tell you why tomorrow. Okay. But before giving, even if it is positive and even if you know everything, you cannot give the same drug to every person. They still require some genetic testing. If you have that knowledge in advance that some people may react badly, you need to test it. If you don't have that knowledge, then you don't have that knowledge, then we will not know in advance. But if you have, you need to check it. Okay. Wow, that is a record. Okay. All right, you guys are off. So we'll all have lunch whenever we have lunch. I'm sure it is not ready as yet, right? But at least we are done. We'll have a break. We'll meet at 2. I'll make sure I come here by 2. Yesterday I got stuck because, there were other people involved. So we'll start at 2 and then we'll go as long as you guys have questions. But do you guys have any questions right now? You have. I'm very glad you have. Go ahead. Mm-hmm, mm-hmm. Yeah, so I don't know about carbonic anhydrase receptor, but lactate acid, which is released, there are obviously those receptors there, and people have tried to inhibit that. So if you inhibit lactic acid secretion, in some way it is good because it prevents it from getting acidified, but that alone does not really change anything for cancer because they have so many alternative pathways. You shut down one, the others take over. So, and also they can also use lactic acid, convert back to pyruvate and feed it to this Krebs cycle there. So, lactic acid inhibition or carbonic anhydrase inhibitor alone is not enough there. The problem is this: you cannot target everything which goes wrong in cancer because normal cells will also not survive. So, you have to go for those guys which are, first of all, they should be ideally different between cancer and normal cells, right? So that only cancer cells are effective. The second very important thing, they should be the drivers. They are the one running the show. Lactic acid secretion is helping, but that is not running the show. You want to go to the culprits who are running the show, who are responsible for lactic acid formation and all this acidification of the environment. You want to go up and go to the master regulator. See, uh, when you are even giving a combination therapy, right, it is considered very toxic because you're hitting two major pathways, right? But here, everything we are weighing against a fatal disease, so we are taking more and more risk, right? Because it's a question of life, right? So, decreasing acidification is good because if you decrease acidification, you can let immune cells come close to it, right? But you know, cancer cells are employing so many ways. They are, you guys, some of you guys, I don't know who was telling me about these exosomes which are released. Somebody, you were telling me, right? So exosomes are really bad. So what are these? These are small vesicles. So cancer cells are releasing these exosomes and they will attack clean, nice neighboring cells who have nothing to do with cancer. And they will have all the bad things in there like RNA and different kinds of stuff, oncometabolites. They will go there, they will make them cancerous, right? So So that is why I say they are using immunotherapy. So immunotherapy is waking up the immune system. So it is going to attack in many different ways, like basically recruiting powerful enemies of tumor. But you know, you are right, you never know. We have to try everything. We have to try everything. Nothing is bad. Maybe using inhibitors of lactic acid in combination with something else is very good, right? So, and I'm sure it has been tested. I am 100% sure. See, so it is there. So we have to test it. And unless you test it, you really don't know what is going to work or not work. There are thousands of targets for cancer. People are testing. Yeah, I know. Yeah. Mm-hmm, mm-hmm. That is becoming very popular these days, photodynamic therapy. Yeah, my friend at Bits Pilani, he's also working on that thing. So yeah, people are trying to treat cancer in 100 different ways, you know. So and it is, it is getting better day by day. Everything is adding up in our arsenal. So we can treat even aggressive cancers these days, right? So we are making huge progress. The only problem is that if cancer cells were not resistant, we would have eradicated the disease by now. Every time we treat them with a new drug, they come up with a new resistance mechanism. So right now in prostate cancer, the worst cancer is neuroendocrine prostate cancer. That did not exist before. This is man-created cancer. They come because we treated them with androgen receptor inhibitors or androgen signaling inhibitors. So that's a constant fight between man and cancer, you know. But I always think that one day we'll discover something so trivial and the cancer will be gone. Like, you don't attack it at the cancer itself, you just switch off something else somewhere. It's like to remove the darkness. Instead of, removing the darkness and throwing darkness out in small pieces, you just turn on the light somewhere, you know. I don't know how, but I'm hoping that one day it will be a dream that this disease doesn't exist. Like, we have eradicated thousands of diseases. Yeah.
I was not able to understand how cancer cells are getting resistant to radiation because it essentially goes and damages the DNA every time you expose it to radiation. How is the resistance? Is it DNA repair enzyme?
Yeah, it is the upregulation of DNA repair enzymes. Radiation resistance is the most common thing. And I had a student some time ago from China. He was a postdoc in my lab. So he's a prostate cancer oncologist and in Wuhan. So he actually works on this radiation therapy resistance. So in that, he has shown that the metabolic pathways are are different in radiation induced cells versus non radiation induced cells, and the metabolites which are formed, they are the one which cause cancer. So when you give radiation therapy, DNA upregulation of DNA repair enzyme, then the metabolism also changes. The enzymes are different between, not in cancer cells versus radiation induced normal cells. I mean radiation treated cancer cells, and so these enzymes behave differently to become resistant. So there is I was trying to tell you, I honestly cannot remember what is that, some setsa or something, it is called CETSA or something. I will check it today and tell you tomorrow. So I know of that person, like I don't know him personally, but I know of him. So he has developed this setsa thing, CETSA or something to that. So there what he does is that he basically takes something which will not even touch normal cells, and it will get disassembled within cancer cells, because cancer cells express an enzyme called alkaline phosphatase in huge amount, and he assembles this whole structure in such a way that if alkaline phosphatase is there, all you do is just pull one trigger and the whole thing will disassemble. Like it is like a whole brick of things going there, like just literally bricks, like, and you just pull out just one piece like house of cards and the whole thing disassembles. So it's like giving what we really want to do is a truckload of bricks on the cancer cells, damage them so badly. This is not molecular mechanism, this and that. You just basically hit them and you die. Don't get up again, right? So I'm hoping this kind of therapy will not lead to resistance, So people are trying all different kinds of things. If I find it today, I will at least include one slide to show you what is that therapy, you know. So people are coming up with really innovative stuff. More questions? Yeah, go ahead. So, so what I know is that they recommend, so I don't think they are targeting microbiota itself in the tumor site, like they're not injecting it there, but what they recommend is to eat healthy probiotic kind of stuff so that you improve the microbiota in general. Um, they are also doing the fecal thing. I don't know exactly how it does, but they are delivering the good microbiota that way also. So the whole idea is to systemically have the better microbiota in the entire body. I am not aware of that they are targeting microbiota directly in the tumor site. See, what I have understood from all of these things, that um, first of all, our knowledge is really incomplete because microbiota did not even exist 10, 15 years ago. This is a new component. So there are so many components of this cancer disease alone that we are completely unaware of, right? So we have less than 1% knowledge, this is my thinking, about cancer, right? So based on every day the knowledge we are gaining, we are changing therapy. But what is miraculous is that, so before I come to the miraculous part, so like there are DNA methylases 1, 2, 3, 4, 5, whatever they are, right? So one is bad, you use inhibitor against one. Maybe one is also doing some good thing, but when you're inhibiting, you are also killing the good part of it, right? Same thing. You cannot specifically inhibit DNMT1 because it has cross reactivity, right? So if this room is full of different problems, we are aware of 1% problem and we are trying to cure that problem. But what is miraculous is that, of course, when the cancer is there, we have to treat it. But the miraculous part is that if we are exercising, eating well, somehow that exercise, whatever the benefits are, they know exactly which one to inhibit, which one to activate. And we have zero knowledge. We don't know what is happening in the body. We are just exercising. And the body actually knows what to turn on, what to turn off. It takes care of every single thing, right? But again, once the disease is there, the body is the one which is actually fighting the disease off. The knowledge that we have gained is that, okay, immune system is shut off. We need to reactivate the immune system, right? So it is extremely important that we gain the knowledge there. So coming back to your question of microbiota, the way they are treating is now systemically the whole entire the cross body. They're trying to improve the microbiota without going individually, because our knowledge is really incomplete in that area. There are trillions of microbes there. We really don't know which one is doing what. What we know is that microbiota is supporting tumor formation, and if it was good, there would no tumor formation. But we don't know at a cellular, at a molecular level, which one is good, which one is bad. Is it making sense? Yeah. And also, many things are circulating in the blood when someone has cancer. So it's affecting other than tumor cells like metabolites, oncometabolites kind of things. Uh, I was talking to a friend, I don't even know who I was talking to. So he told me a very funny thing. He said if you measure the level, yeah, I remember whom I was talking to. He's a person of Indian origin who lives in US, and he said that if you measure glutamine levels in bloodstream, you can predict Alzheimer's disease 5 years before it shows its first symptom, right? So I'm sure that we don't have knowledge, but there are things which are happening. There are changes in our bloodstream, changes in something, and if we know exactly to what to look for, we can predict the disease long before it manifests in our body. We have some knowledge about few things, right? So, but cleaning it systemically, if it is not toxic to the body, is good. Microbiota is one of those things, eating prebiotics. So they don't have to go to the tumor site. They can just clean up the whole body, right? But there is no doubt about it that the microbiota at the tumor site is pro-tumorigenic. Go ahead. Say that again. Oncolytic viruses, yeah. Yeah, so what are oncolytic viruses is that um, huh? Yeah, so these viruses, I'm just trying to remember what is there inside because I am thinking about oncolytic vaccine versus oncolytic viruses. So so what happens is that the viruses which are there, so viruses are used as a vehicle to deliver the things to the tumor site to basically kill the tumors. Okay, so you know what is an oncolytic virus? So what is a vaccine, cancer vaccine? You know, right? So like many of you may have used lentivirus or retrovirus in your lab. I don't know if you have used. You have not used, right? So viruses are very useful as delivery vehicles. Okay, so oncolytic viruses are those viruses. So viruses, you have seen this kind of viral structure, right? So you package your material inside the virus, and virus can very easily go to the site, and if you decorate the virus with the right thing, it will go to the tumor site. Okay, and then it will release the cargo, and that cargo will kill the tumor cells. This is a very generic thing. Oncolytic virus will come in details when I will tell you about biologics tomorrow. So I will tell you exactly what it is, but this is a very simple version of it. And cancer vaccine, you know, the antigen against specific towards cancer. And so the immune response will be triggered, which will kill the tumor cells. So tomorrow I will show you a slide about biologics also. Biologics are like antibody-based therapy or cancer vaccine or oncolytic viruses or CAR T therapy. All of these things will come tomorrow. So small molecules we know very well, right? Drugs. But these guys will come. There are 9 topics we'll discuss: fusion proteins, things like that. See, we are finishing up even the tutorial right now. Then we can really enjoy in the afternoon. Oh, absolutely. You are going to get every single slide. This morning I woke up at 4, not 4, 2. So I sent all the slides to Venkatesh already in advance. Yeah, so you will get everything, every single thing. I modified a lot of things this morning, and so yeah, you will. I have already sent him all the slides for tomorrow also. So he will send you everything. Uh, you had a question, yes, go ahead.
MYC is part of the signal transduction pathway. So it is a protein that it will be only active when the signal molecule is binded to the receptor.
You're talking about MYC.
MYC.
Yeah.
Right. So it was part of the signal transduction pathway.
So here is the thing. All the proteins are part of signaling things. Nobody exists in isolation. They're all part of some cascade. So go ahead.
Generally, proteins that are part of signal transduction pathway, specifically they are only in active form when a signal molecule is binded to that specific.
So let me correct you here, right? Let me correct something here. All the proteins, none of the proteins are like one protein living in isolation. Every single thing is part of something. They are part of some signaling cascade. They can be top of the ladder, they can be the bottom of the ladder. So top of the ladder is receptor, which is starting the signal, and then it is going to the nucleus, and all of these things are in between, right? And nuclear proteins are there and things, and then the response comes in. So MYC is also part of a signaling cascade, right? But what happens is that MYC is a very unstable protein, okay? But And so I was doing gene chip analysis when it was developed back in early 2000, and I could not detect MYC because MYC just degrades so quickly, right? It is a very unstable protein. And there was a big controversy going on at MYC levels, and we had developed this approach. We could have answered that. But even then, I could not even isolate MYC because of the unstable thing, right? But in cancer, MYC is really stabilized. It is really stabilized. So what happens is this, what goes wrong? So you guys really answered a lot of very good questions today. So think about DNA makes RNA, RNA makes protein, right? So sometimes the problem is at the DNA level, there is gene amplification. So every time you will make more RNA. Then sometimes the problem is at the RNA level, that there are microRNAs which will, even the RNA levels are same, they will degrade it, so it is no longer made, tumor suppressor is not there. Or they will increase the translation, more is this, or they will stabilize it, or they will destabilize it. So even if you make the RNA, you don't know how much protein you are going to make. Now let's come to the protein, and that brings me to MYC. So when MYC protein is made, it is degraded very quickly. Okay, but in cancer, MYC is not degraded because it binds to other proteins. And when it binds to other protein, it is extremely stable, like really, really stable. And then, so when you look at protein levels in the cell, this protein level, this steady state protein level that you're seeing, that is sum of synthesis and degradation, synthesis and degradation. But if you stop degradation, the levels will keep on increasing, right? Makes sense? So MYC is really stabilized in cancer. Okay, and usually when it is in partner with its partner, I work with that protein, but right now it is not even coming in my head what is the binding partner. I think it is aurora kinase. So when it binds there, MYC is also highly active. Okay, so sometimes this is a very common theme in cells, that if there is huge excess of protein, they are very active also. They don't degrade. They are very obviously they are very stable, but they also become highly active. Okay, and their conformational changes which keeps them in active state. So to make a long story short, MYC become independent of outside signal. When it is supposed to be degraded, it is not degraded. Okay, and like somebody told, who said about localization today? Somebody said about localization today, right? So one of the biggest story in RAS signaling is that RAS is probably one oncogene which is expressed in all human cancer, and it alone is responsible for more than 30% of human cancers, correct? And RAS is a GTP binding protein, and GTP binding to RAS is so tight that nobody in the world could ever think that they could remove GTP from there, because it's picomolar binding, extremely tight, right? So when I started my lab at Purdue, so we were working with G proteins, which is RAS was one of them. So we developed a method there, and so I know a lot about the tight binding, and we could never do. But then the first time ever, one person actually developed an inhibitor which can kick GTP out of the pocket, and that person was my postdoc advisor's lab. Okay, so in his lab, what they did, they looked at the crystal structure of that, and so this is the active site, and this is where GTP binds. And remember, we were talking about allosteric sites yesterday. So he found that there is a small pocket allosteric. So he just made an inhibitor and accessed that pocket, and he could kick GTP out. And that was one of the biggest discoveries of our time. So, and he has opened a company, but since he has found, now people know there's allosteric site. So there are many, many inhibitors there, like many inhibitors. His first inhibitor is his, and he has a company, and I am sure it is going to clinical trials. Hi. So we have finished all the classes for today, and tutorial is going on. So yeah, really ahead of time. So everyone has afternoon off today. We are really. Huh? 4 lectures. One after the other, no break. Because there was nobody to tell us to take a break. So we went on, we rely on you to tell us it's break time. You were not there, we didn't know what to do. We knew lunch would not be ready. Let's use this time and then have a fun afternoon. Yeah, so coming back to that, now there are many inhibitors for that RAS protein. So, but before that, when we did not know, then we were not allowing it to go to the membrane, because if you don't allow it to go to the membrane, it is ineffective. So what you were saying, that it should not translocate there. If you can find translocation inhibitor, it will not be effective. So that is a very good strategy also, right? Translocation inhibitors. This has to close before I can tell. बंद कर दिया. So this story is of Peter Schultz. He's one of the biggest scientists of our time, and he's my postdoc advisor's advisor, right? So he's my academic grandfather. So that's why I know all the internal stories. This is not in public domain. So he's the one who developed RNA chips. So he was sitting with few of his friends, and this is a very famous thing. So somebody said, we could possibly make DNA chips and we could possibly make RNA, and this is just few friends having drinks and chatting. So Peter Schultz said, oh no, it is going to bind everywhere. It's not going to be specific. And then he was the one, along with these 3-4 guys, who developed that. And they opened this company called Affymetrix. If you go to his Google, you will get the whole name. And they opened it, and it became $1 billion company, and then they sold it. So when Pete was working, so I worked at Novartis, you guys know. So Pete was the director there, and Pete's office was there, and my office was next to Pete's office, closest to the boss office. And he used to come at 4 a.m. in the morning every single day. Okay. And before coming to 4 a.m., he used to run and then be in the office at 4 a.m. every single day. So those of you, in US, you have different time zones, right? Like East Coast, then, 3 hours difference. So he was at UC Berkeley initially. So UC Berkeley is in Pacific Coast. We were at Princeton, right? So Princeton is 3 hours earlier. So if this is 9 a.m. there, it is 6 a.m. there. So when my postdoc advisor was his student, so when he had to talk to Pete, we were at Princeton. He had to come early to find Pete, because Pete was at Pacific Coast at 4 a.m., but that would be 7 a.m. here. And my boss was not here at 7 a.m. So he had to run to come at 7 a.m. to talk to Pete at 4 a.m. because after 5 a.m. Pete was inaccessible. He was so busy. So it was very funny. So he is the one, he said it is not going to work, and then he is the one who developed this Affymetrix chip. So while I was there, one day we came to know Pete sold that thing, and just he got 32 million. So I took a peek at Pete that this morning he made $32 million. This is back in 1999. It was a big amount then. Pete was just signing the paper like this, like it is no big deal. So that's the story. So like today we may say, oh, translocation inhibitors are not that good, but tomorrow you will make so much money out of this thing, right? You never know, because this is how ideas are developed. You start throwing wild ideas, and it may seem wild idea today, but it may turn out to be actually the coolest idea, All right.